Fibrosis Research and Translation. He is also the LaConte Chair in Cardiovascular Research, University of Colorado, and is honored with 27 awards, besides being ad hoc reviewer, scientific advisory board member, and also he is in several journal editorial boards. It's an honor to welcome Dr. Timothy A.
All right.
Good.
Can you hear me? All right.
Yes. So good afternoon, everyone. My name is Pilar Alcaide. I'm going to be moderating this session with Dr. Hien Lat from His main amazing discoveries in the field of epigenetics and cardiac fibrosis, and we're looking forward to hearing from you, Tim.
Well, thank you, Pilar, and thank you, everyone, for being here. I know it's a little noisy as everyone gets settled in. Could you bring my slides up, please? All right. I want to start by thanking the leadership of the PhD Institute, PSG. This has been an amazing experience. Namaste. I really appreciate this. And thank you to the Saktis, the local Sakti, and my boss, the leader of our in South the United States in the scientific community. So Denver is located in the western part of the United States, shown here. For those of you who don't know, the East Coast is over there, and the pointer's not working, so you'll just have to take my word for it. But New York is over here. Yeah, there we go. California is over here, and there's Denver. And Denver is best known for being part of the Rocky Mountain system. This is not as dramatic as the Himalaya system, but the Rocky Mountains are a beautiful place, especially if you like to hike and camp and ski and things like that. So the title of my talk has this word in it called epigenetics, so I thought I would spend a little bit of time describing generally what that means. This is a depiction of a cell, and as all of you know, cells have a cytoplasm and also a nucleus. If there's a better pointer, I'd like that too, if you have one. So within the nucleus is DNA, as everyone knows, and that DNA is transcribed into messenger RNA, which then becomes protein. This is a very important process. Now, DNA depicted here, if you were to take a single cell from your that regulate epigenetics, and they're loosely referred to as writers, erasers, or readers. I'll be talking a lot today about erasers. These are proteins, they're enzymes that remove chemical marks from DNA and proteins to regulate gene expression. These are epigenetic regulators by doing so. All right, before I dive into the science, Sakti from Cincinnati asked me and others to talk about ourselves. This is challenging because I don't like to talk about myself, but I'm going to spend a few minutes doing this. And I want to preface this by saying that my background is vastly different than the backgrounds of all of you and of anyone else in the audience. But I think that there are some lessons that I've learned along the way that apply to everyone in science, and I would like to tell you a little bit about those lessons. And I think they apply to teachers and faculty members as well as students. So this is where I grew up. It's called Springfield, Missouri. It's about 1,000 miles away from Colorado. And it's a small town of about 150,000 people. I had a wonderful childhood. I still have a lot of friends from my early days in Springfield, so I loved it. It's a very, we call it a homogeneous community. There's not a lot of diversity there, and that's one of the downsides of growing up there. The most famous person to emerge from Springfield, Missouri is not me. It's Brad Pitt, the movie star. I did not know Brad, but I knew his sister, and she told me that he was dropping out of the university to go pursue a career in acting, and I thought, wow, this guy really doesn't know what he's doing. He's a foolish person, but he's been successful. So he's the most famous person from my small town. Now, I attended the University of Missouri, about a 3.5-hour drive from Springfield. And to be honest with you, I didn't know what research was. I didn't hear about it in Springfield. I didn't ever hear the terms basic science research. And I went through 3 years of college without knowing anything about it either, because back then, the university didn't do a great job of exposing students to research opportunities. And I thought that research was the same as the research class, the laboratory classes that we were having, and I didn't really enjoy those, and I wasn't very good at them either. But in my 4th year of college, I had a professor, he's shown here, Stephen Alexander. He has no idea who I am, but he had a major impact on my life, because instead of assigning us a textbook to read about science, he assigned us papers, original research articles. And that was really eye-opening for me. It was the first time when I started to realize what research is, and it got me extremely excited about the potential of a career as a scientist. So I decided at that point that I wanted to work in a lab at the university. So I started knocking on laboratory doors, talking to professors and trying to get a job, something as simple as washing dishes. And no one would hire me because I didn't have any research experience. It was very frustrating. So on the 6th door that I knocked on, I went 5 times, and there was this guy, and he's still around. His name's Mark Hannock. He's a really rigorous, great scientist. He's not well known to you, but he also had a major impact on my career because he said, sure, you can work in the lab, but I'm going to treat you like a graduate student so that you can understand what you're getting into. And that's what he did. And Mark had just finished training with a Nobel Prize winner from the University of Wisconsin, Howard Temin, who co-discovered reverse transcriptase. And so Mark was just starting his lab, and he was very aggressively pursuing the science. And that was a good thing for me because it taught me what a very rigorous science is all about. The problem is that, okay, so this is my first project. I'm going to take a show of hands. How many of you know what PCR is or have heard of that? Polymerase chain reaction? So quite a few, not everyone. Fewer than I expected, but it's a technique that is widely used to amplify DNA. And when I first started in science, it was a new technique, and my job was to clone a gene using PCR. And my job was to clone a gene from chickens called CREL. So I got to cut heads off of chickens, and I isolated chicken spleens and then RNA. And I was to amplify the CREL gene and clone it into a vector. That was my job. So I spent about 2 months trying PCR, and nothing worked. Then I went back to Mark, and I said, I don't think this technique is valid. He said, keep trying. And this, for those of you who have done PCR, you know that a lot of times we use pipette tips that have filters in them to prevent contamination. Well, this was before those tips existed. So after about 3 months, I finally got PCR to work. There was a nice band, amplified DNA band. Then what I had to do was to sequence that DNA that I had amplified using these gels. And that took another couple of months because I could not pour a gel without a bubble in it. But then I did, and ran the gel, the sequencing gel, and what you did back in those days is you put plastic wrap over the gel, and then you would dry it and expose it to film, X-ray film. Well, I forgot to put that plastic wrap on the gel, and I put it on the dryer, and the thing just disintegrated. So this was my exposure. This is my early experience in science. And Mark pulled me to the side. It was a Monday, and he pulled me into his office. He said, Tim, I appreciate you, but you might want to think about doing something else because I was so bad in the lab. But that was really an eye-opener for me because even though everything I was doing in the research lab was failing, I knew that I loved it, and I wanted to do that as a career. And when Mark had that, we call it a heart-to-heart talk, but that's what we call it in the United States. And when he had that difficult discussion with me, everything changed. And I think what I realized at that point is that I was about to lose something that I wanted to do, and so I took it even more seriously, and that really changed my career. So I thank Mark for having that difficult conversation with me, and that's a lesson that I try to apply as a professor. Sometimes it's hard to tell people that they're not doing a good job, but in the end, if you can have that difficult conversation with them, it will help them ultimately. So after Mark's lab, I had a lot of opportunities to do graduate work and to get a PhD. And I went to Vanderbilt University here. It's in Nashville, Tennessee. It's a beautiful place. Dr. Arthur Holly went there as well. And I was also asked to show a picture of myself, so I found one. And I'm only showing it so that my friends in the front row will know that I once had hair on the top of my head. There it is. So that's me at Vanderbilt. All right. So the lessons here are you need to be persistent. Don't give up. Science is very hard. You need to keep pushing. Don't let the negative things, the failures, get in the way of your dreams. You need to be passionate about it, and I realize that not everyone loves science, and you need to recognize whether or not you love it early and decide to pursue it. If you don't love it, do something else, because I think you'll be miserable as a scientist if you don't enjoy it. And then you have to be lucky. I've been so lucky. I had these mentors show me the way. I was born in a country that has a lot of resources. I was born into a family that was supportive and loving and valued education and had the means to send me to a university. So I recognize that I've been very lucky along the way. I'm also lucky because, and this is not a good thing, I'm a white man. And in the United States, unfortunately, white men tend to do better and have more opportunities than others. And we're trying to change that and support other diverse people and women and minorities so that they can be successful scientists too. And this is something we're actively trying to overcome in the US. So with all that being said, I now want to get into a little bit of science. I want to keep it very general, and I really want someone to ask me a question after this talk, because there's nothing worse than being a speaker and not receiving any questions. So please keep that in mind. You've already heard about heart failure from the esteemed speakers who came before me. And this is, I think, a really eye-opening picture. This is a surgeon holding up a heart, a normal heart, and a failing heart. And you can see that the failing heart is much bigger than the normal heart. And this normal heart is getting ready to be transplanted into the individual from whom the big heart was taken. We call this pathological cardiac remodeling. What that really means is that there's changes that are occurring in the heart that change its structure. And two of the hallmark changes that are associated with heart failure have already been introduced. These are cardiomyocyte hypertrophy and cardiac fibrosis. Cardiac myocytes are like neurons. They're post-mitotic. They don't proliferate. So when they encounter stress, they either do one of two things. They'll either grow through hypertrophy to overcome that stress, or they'll die. And both things, while hypertrophy is initially a beneficial adaptive response that allows the heart to overcome stress, long-term hypertrophy is bad and leads to heart failure. And you can see here, this is a scanning electron micrograph. These are micrographs, and these holes are where the cardiomyocytes were. And so you saw the small heart and the big heart that I showed. They were cut, they were sectioned, they were cut in half and cut into very thin sections before performing this procedure. And you can clearly see the enlargement of the cardiomyocytes, and then the stuff between the cardiomyocytes is extracellular matrix. We heard beautifully from Dr. Lowell in the last presentation about what fibrosis is. This is scarring. And what happens is when you have hypertrophy and scarring of the heart, it can't pump and relax like it should. And that leads to heart failure and ultimately death. So we've worked hard in the lab to try to create models of hypertrophy and fibrosis in a dish, meaning in a Petri dish. But in this case, the Petri dish is a little bit different. It's called a 96 or a 384 well plate. So it's about the same size as a regular Petri dish, but it has compartments to put cells. It has 384 compartments to put cells. And we use this to do something called high-throughput screening. So I'm only going to talk about our fibrosis in a dish work, not the hypertrophy in a dish work. The way this assay or the system works is that we use cells called fibroblasts. These are the major cells that create fibrosis and scarring of the heart. So we isolate the fibroblasts from hearts, and we put them in those 96 and 384 well plates, and then we screen for chemicals that block the conversion of those fibroblasts into an activated state. Those activated cells are sometimes called myofibroblasts, and they produce a lot of the material that results in scarring of the heart. So we did this using fibroblasts from the heart and the lung and the kidney, and we screened our chemical libraries for chemicals that could block this activation. And we measured the activation using the high-throughput microscope shown here. So this is a microscope that goes around and takes pictures of the cells very rapidly, and then it provides a quantitative assessment of markers of fibroblast activation. And here's an example of a marker. You can see these green cells, those are activated. And we're looking for chemicals that block their activation based on that green signal. So, this is a chemical library. It just means that we have thousands of chemicals in these plates that we keep in the freezer. And then we use robots to move those chemicals onto the cells. And then we screen for compounds with the ability to block activation of the fibroblasts, because we believe that those compounds will be able to block fibrosis, which is a key component of heart failure. So now we're back to epigenetics. I told you about these chemical marks that occur on proteins and DNA that regulate mRNA synthesis. And one of those chemical marks is called acetylation, which I've abbreviated here as AC. And it's regulated by writers, readers, and erasers. And in our high-throughput screen of all of those chemicals, we found many of the hits, many of the chemicals that blocked fibroblast activation are inhibitors of the histone deacetylases, HDACs, which are erasers of this acetylation mark. And here's a picture of cells treated with an HDAC inhibitor. And the blue, those are nuclei, and then in the green is the marker of fibroblast activation. And you can see that there are plenty of nuclei in the cells treated with the HDAC inhibitor, but the fibroblast activation is gone. So we teamed up with a company in Italy called Italfarmaco, and we tested this compound called givinostat for its ability to block fibrosis in an animal model of heart failure. The way givinostat works is that it sits down on the HDAC enzymes and it chelates a zinc atom that those enzymes need for catalytic activity. This is a very interesting compound. It's been tested in children with Duchenne muscular dystrophy, and it's awaiting FDA approval for that indication. It's also nice because it's a very potent HDAC inhibitor, and it's orally bioavailable, which means that you can swallow it, and then it gets into your bloodstream and goes where it needs to go. And ultimately, we would like to develop a new pill to treat heart failure, so that's a real benefit. So this model was introduced earlier by Pilar and Hind, and it's called the transverse aortic constriction model. It's where we take mice and we open up their chest, and then we tie a suture around the aorta, and that creates a pressure overload. It's like mimicking hypertension or aortic stenosis or valve disease, and it puts a pressure overload on the heart so that the heart then grows, undergoes fibrosis, and then ultimately fails. So when we did that, and then we took the hearts out and we sectioned them to look at fibrosis, this is what we found. Now here, the red signal is scarring. And you can see that the TAC surgery caused scarring of the heart of these mice that was blocked by the HDAC inhibitor givinostat. So we firmly believe that these epigenetic regulators, HDACs, regulate fibroblast activation and fibrosis of the heart, and they could be used to treat heart failure by blocking the scarring of the heart that is associated with heart failure and pathological cardiac remodeling. So I want to end with 2 slides. I love what I do. The reason I put job in quotation marks is that I don't even consider this to be a job. It's really a passion and something I would probably pay someone to do. It's really fun. Why is it so much fun? Well, we're explorers. We get to come in every day with the potential to make discoveries, and that's really exciting. We are storytellers. We're teachers. We're artists. We get to present our work and use artistic skill to try to convey a message. We get to work as part of a team. And we also have the potential, our discoveries have the potential to help millions of people who suffer from cardiovascular disease. So I just think that being a scientist is a wonderful profession, and I would encourage any of you who are passionate about science to continue to pursue it. And then finally, I want to end with this, and this is actually the best thing about being a scientist. I'm standing here in southern India in front of all of you. I'm from a small town in Missouri that I mentioned is not very diverse. But because of science, I have friends all over the world. Many of them are here on stage and in the front row, and I get to travel places and talk about science. And I didn't realize when I was going to Mark's lab to start my career in science that this would be the case. But this is just a really fulfilling element of being a scientist. So with that, I'll close, and I'll be happy to take any questions. We have a great group here in Colorado. I do want to highlight Rashida Bagchi, who did her master's training at the University of Baroda in India before moving to Canada to get a PhD, and then she did a postdoc with us in Colorado. So I will end there, and I'd be happy to entertain any questions. Thank you.
Well, this is open for questions. As Dr. McKinsey said, there's no worse thing in the world than giving a presentation and not having any questions. So I encourage all students to get to the microphone and ask a good question. As he was perfectly on time, and there's time for them.
Wonderful. Your message finally about the science and the scientific community is said to be something exemplary and which creates a big type of your the awareness among the young students who are assembled here. My congratulations for that. And I have two general questions, which is not exactly related to their presentation, but you know that the cardiac disorders, there is a genetic predisposition that is normally being seen. So in the US, maybe in India, you can find that still it is in a nascent level, and it has to be taken up in the next level to be taken up. But in US, what is the condition in the case of your detection, in the case of your predisposition to cardiac problems in terms of genetic level? Then my second question is that, how far the gene therapy is said to be having a success story in preventing and treating the cardiac disorders in the case of your US scenario?
Okay, the first question relates to genetic predisposition to epigenetic changes that cause heart failure, if I understood you correctly. And there's a lot of evidence for this, primarily based on mutations or polymorphisms that are occurring in regions of DNA called enhancers. These are elements of DNA that are not encoded into mRNA and proteins, but they regulate the expression of genes. And as we dig more deeply into human genetic data, we're realizing that there are many mutations occurring in those enhancer regions that are influencing transcription factor binding and epigenetic events that ultimately trigger the heart failure process. It's in a nascent period, as you mentioned. We're just now developing the technologies that allow us to determine how epigenetics are controlling human heart failure. So it's a very exciting time to be in the field, and I think there's a lot of potential for epigenetic regulators in the heart failure process. In terms of gene therapy, I am not an expert, but I am sort of following the field. And what you're going to see, I believe, is gene therapy for rare forms of heart failure, monogenic diseases, rather than the forms of heart failure that I was trying to describe here, which are due to, they're acquired through hypertension, myocardial infarction. I do think you're going to see some breakthroughs within the next 5 years that are related to genetic cardiomyopathies that can be treated with adeno-associated virus encoded proteins to treat the disease.
Thank you. Thank you.
Yeah. So there are 5 FDA-approved HDAC inhibitors, all for the treatment of cancer. This is one of the reasons that we're so excited by the results from Ital Pharmaco, where they treated children, boys with Duchenne muscular dystrophy, and they showed a benefit in those boys. And remarkably, the compound was well tolerated by the boys. So this would be the first non-oncology indication for which an HDAC inhibitor has been approved, which would open the door for testing HDAC inhibitors for other non-oncology indications such as heart failure.
One more question and quick answer.
Thank you so much for your wonderful session, sir. It is very motivating for a student like me. And my question is, could you please explain a little bit detail how to identify the fibroblast activation, sir?
Yes, that's a great question. The way we identify it is to use antibodies and immunofluorescence staining. So we have antibodies that recognize certain proteins that are only produced in activated fibroblasts. The example I showed here is for a protein called alpha smooth muscle actin. In a quiescent or a resting fibroblast, that protein is not expressed, but when the cell gets activated by a pathological stress, that protein comes on, and we can detect it using antibodies and fluorescent secondary antibodies to stain specifically that protein.
Thank you so much, sir.
Thank you. Thanks.
I invite Dr. B. Ramesh, Associate Professor from the Department of Biochemistry, to felicitate Dr. McKinsey.
Thank you, dignitaries. We are privileged to introduce the renowned speaker for session 6, Dr. Shyam Bansal. Dr. Shyam S. Bansal is the Vice Chair for Basic Science Research and Associate Professor at the Heart and Cardiovascular Institute, Pennsylvania State University. He started his journey as a BS candidate in India and obtained his PhD from University of Louisville and postdoctoral fellowship from the University of Alabama, Birmingham. His active and completed research projects were funded by the National Heart, Lung, and Blood Institute, Ohio State University, and the American Heart Association. He has received 11 plus awards and possesses 2,535 citations with an ITIN index of 37 for 45 plus publications and 4 book chapters. His research and teaching expertise has earned him several certifications and memberships, including the editorial board member in renowned journals, and his works have been featured in the Time magazine. It's our honor to welcome Dr. Shyam Bansal to share his expertise on the topic immune dysregulation and cardiovascular diseases. We kindly request Dr. P. Shaktivel Sariyappan and Dr. Pilar Alcade to chair and moderate the session.
So I'm pleased to introduce Dr. Bansal. Everything was told about how his career trajectory and how successful he is to this date in the United States. I have to say that working in the same field, he's also a fantastic colleague who's actually helped a lot during our stay in India as well. So we're looking forward to your talk, immune dysregulation and cardiovascular diseases.
Thank you, Pilar. Can you hear me? Okay. Hello, everyone. I would like to begin by thanking moderators and organizers for this conference and the leadership team of PSG College of Arts and Science for bringing us all here and giving us this platform to talk to you all. It's really a pleasure to be here. As you can see, I didn't write any of my affiliations here because none of that defines me. What defines me is that I'm a teacher, right? I chose to do my PhD because I wanted to be a teacher since the beginning. And I'm a scientist because I am curious by nature, right? As you will see, I always loved to read. And when I say I love to read, it's not that I love to read science, right? I actually used to fail in science when I was in my 8th grade, right? I just loved to read anything that can gain my attention, right? And that's what dictated all of my life, as you will see slowly. Okay, it's not moving. Okay, it's too much. It's slow. Okay, so I was born in a small village in Punjab. My village was half a kilometer of stretch of a road, where buses will just come and stop for a minute or two and then go, right? I was one of 4 siblings, and as you can see, this is one of the occasions of Raksha Bandhan, where my sisters and we were sitting together. And if you'll pay attention, you see this, right? There is a book in my hand. So even though we were celebrating Raksha Bandhan, I was reading a book there, probably a story book. I can't identify what kind of book is this now, but I can tell you it's a story book. I loved reading comics book, right? Who reads comic books here? Right? Anyone? Nagraj, Bhokal, right? Those were the ones that I grew up reading, and I was crazy about them. My father always hated, but I'll still read them, you know, hiding things from him that way. I was born in Punjab, and I was born at the height of terrorism in Punjab. My birth is 82, and 84 is when Indira Gandhi was assassinated. And you know how the Punjab was so worse at that time. My father, after that, moved to Delhi so my father moved to Delhi in late 1980s for better opportunities. If you're wondering where I am in this one, I'm here. This is me. That's my 6th grade picture from here. So my father moved to Delhi in 1980s for better opportunities. And I was still with my grandparents in village. And in 1991, when my grandfather died, my father took me to Delhi, and that's where I studied for most of my part. I studied in a government school, Sarvodaya Bal Vidyalaya. I did my 11th and 12th from there, and then followed by Delhi University. That's where I did my B Pharmacy. Like everyone else, right? I had science and biology in my high school. I wanted to be a doctor, right? I wanted to give DPMT exams. I did give DPMT exams after my 12th grade. That's what it was called at that time. I know the name is different now. I got maybe 700 rank. I was like, maybe I'm going to prepare for 1 extra year, but take admission in a college. So I ended up taking admission in pharmacy, and I started reading it. The moment I started reading about pharmacy, I just loved it, right? And I got so engrossed with pharmacy that I didn't even give exams for medical school after that. I just made that drug development process as my passion. And you will see how I have utilized that training as I moved up or did things. Right? As I told you, right, I like stories. I like books, right? I'm going to try to take you all like a story, right? What we did, how we are doing. I love comic books, so you will see a little bit of comic book here as well, right? A comic book that we wrote and working on it still. So that's me. It worked now. Right. Okay, so what is heart failure, right? It's 2nd stage after heart attack, right? Several of us told you, right? Heart attack is what? When blood supply to the heart stops, right? Something happens, something goes and blocks your coronary artery, and blood supply to a part of heart stops. And that leads to heart attack. That's when we feel pain, and then we go to doctors, and if we are lucky, they are able to remove that blockage in time and then send us back home. But in several patients, if they don't go to the doctor or they don't realize it happened, they slowly develop into a 2nd stage of heart attack. We call it heart failure, right? What happens in heart failure is heart grows bigger and bigger and does not pump out blood adequately, right? And this is what heart is doing. So if there is no oxygen to the heart due to stoppage of blood supply, right, it's going to die, right? So cells below that stoppage die. So 30% cells die, 40% cells die, and depending upon that, your heart has a dysfunction now. So what happens is, in a normal heart, it pumps around 60 to 65% of blood every beat, right? Say if 100 ml of blood comes to the heart in 1 beat, it pumps out 60 to 65 ml, right? And that's how it maintains adequate function. When we say failing heart, it doesn't mean that the patient is dead, right? It's a heart failure, yes, but patient is not dead. It only means that heart function has dropped below 45%, right? It's like if you don't get 33% max, right, you fail, right? That's exactly what it is. If heart does not have more than 45% function, we say it's in a state of heart failure. And we try to identify reasons why it happens, right? Why heart keeps on decreasing its function with time in patients. So what happens is, whenever blood supply stops to the heart, cardiomyocytes, the cells that we have been talking about, they start dying, right? When they start dying, it induces an immune response, right? Our heart sends a signal to the body hey, I'm hurt, I'm bad, can you send me some help? And that help comes through immune cells. Think about it this way, right? We get injury on our skin. What happens? We get a cut, and then we see a plug formation, right? And then blood stops, and then slowly that skin heals in 2 weeks, right? Same thing happens with heart. We had an injury, heart says, oh, send me help, and the same immune cells that can heal your skin come to the heart. They try to heal the heart, right? So they come there, they try to make a patch, right? The patch on the skin wound that we see, that's all fibroblasts, as Dr. Lal already described, right? And same thing happens with the heart. Those immune cells come there, they clear up whatever dead tissue is there, whatever they find is not required, and then they patch it up like a Band-Aid. That's called fibers, right? They make a fibrotic scar on top of it, and then immune system goes away, right? If you look at your skin wound, right? If you get an injury, after 2 weeks, you don't even see a scar. That's what happens, because immune cells have gone back. They have healed the surface. Same thing happens in heart. They patched the heart, and then they go back after 2 weeks, right? That's what happens in a normal immune wound healing response. Now, what are immune cells, right? So our body during evolution created several ways to save us, right? You can say God made these or nature made these. What happens is we have a lot of bacteria, viruses, and fungi, mold floating around us, right? But we don't get sick normally because we are protected. We are protected because of our skin. Our skin doesn't get anything go inside us, right? Our stomach, if anything goes inside, the acid, it destroys it most of the time. So that's our first line of defense, right? That's where we have the first line of defense, our skin, our mucous membrane, that protects us. But if we have an injury on skin, bacteria can go inside, right? Now, bacteria is inside, virus is inside. So how do we fight that? That's where our second line of defense comes in, where immune cells get activated, right? There are these immune cells, they eat up those bacteria and do different things so as to get rid of those phagocytes. That's second line of defense. And the third line of defense is T cells. T cells are, I would say, most potent immune cell in a way that one wrong T cell is enough to kill a whole human being. How? Autoimmune diseases, right? An autoimmune disease starts with one bad T cell that gets activated against your own body. And we have no cure for that. And then person either keep T cells suppressed or they die if they don't do that, right? So that's what happens. So T cell mediated immune responses are the strongest immune responses that we can see. So what happens, how an immune system works? As I said, a bacteria, a virus, a pathogen goes inside our body. Then the innate immune system we call phagocytes, right? They come in, they eat up. They eat those bacteria, they engulf it. And then they chew it up, make smaller proteins out of it. And then they bring those smaller proteins, as shown in these green dots and red dots, and then they bring them on their surface. Then T cells go and read those chewed up proteins as signals. And if that T cell is able to bind with it, it gets activated and then induces an immune response, right? That's how T cell activation takes place. So what are T cells now, right? As I said, anywhere, wherever we require a very strong and potent immune response, monocytes, macrophages, dendritic cells, the so-called phagocytes, they eat up those activate T cells. T cells come in there and then they are like Superman, like superheroes. They shoot cytokines, chemokines, proteins. And these proteins then either activate more phagocytes or they do things that can stop those bacteria or virus from breeding, from proliferating more, right? And that's how they keep those bacteria and viruses away from our body. So what happens in tissue damage? This is typically when if your skin gets injured, for example, right? Your skin gets injured, bacteria gets access to our blood supply, to our inner tissues where we have no protection. In that case, phagocytes come in there, they eat up those bacteria, present it to T cells, and then they clear up those bacteria from that side. Once that cleared up, the immune cells get activated, they go in there, and then they patch it up, right? So this is what happens in a normal immune response. And it happens actually in every wound. So how can we study heart attack and heart failure in lab, right? We cannot give heart attack to humans and then study it, right? So what we do is we use mice. Our subjects are these mice here, right? So what we can do is, so we can give them heart failure. How? So I have a video here to show. This is an echocardiography video of a normal beating heart in a mouse. This is the left ventricle, and you can see it has almost 60 to 70% contraction. That's the ejection fraction that it's pumping out blood. And what you can do is you can tie its coronary artery as it's tied here. And when you do that, these animals slowly get into a phase of heart failure, shown here, right? So look at these hearts, right? They are so big, they are huge. They have huge systolic dysfunction, and they are not beating properly, right? The beating is only 20 to 30% in this. So that's a characteristic of heart failure. Systolic dysfunction is a big deal. So this is how we study heart failure in lab. We take these mice, give them heart failure, and then we study what's going on with T cells in them. So when we looked at this, so we gave them heart failure and looked at immune cells, we can see there are many immune cells during heart failure in these hearts, right? As you can see here, these are just flow plot just to see, so we can label them with CD45. This is a protein that we can check, and every immune cell has this protein, and we know what kind of, how many cells are there. And reason being, because whenever there is damage, immune cells can just follow whatever is released by the heart, and then they extravasate into the heart. They should not be in the heart, right, ideally. So whenever we have heart attack or myocardial infarction, we have cardiac injury and cell death. And that cell death induces immune activation and the recruitment to the heart, right? But the question is, where are those immune cells coming from? Right? They must be coming from somewhere, right, in our body. And if you look at spleen that way, so you see they're coming from spleen. This is 1 day after heart attack, right? So spleen loses 1/3 of its weight, right? It's not that the cells are dying out of spleen, because a lot of spleen stores 99%. Spleen is composed of 99% immune cells, right? So these immune cells, they leave spleen and then they go to the heart after heart attack. So we know they are coming from spleen and bone marrow. Others have shown bone marrow is one other source for these. So whenever we have heart attack, cardiomyocytes, they get injured, they die. And when they are dying or injuring, they release proteins like chemokines and cytokines, just like a cytokine storm. These proteins and cytokines, they get into bloodstream, blood circulation, and then they go to tissues like spleen and bone marrow. They go there, and then they activate immune system. They activate monocytes, macrophages, and these monocytes and macrophages then go to blood. And through the blood, they then follow these gradients and then go to the heart, right? So this is what happens during heart attack and heart failure. But what happens during heart failure now is, so heart is patched, right? But 20 to 30% of heart is already dead, right? It only takes 3 minutes of time, right, without oxygen for us to die, right? So the same thing happens if cells don't get oxygen, enough oxygen for a long time, those cells die and their cardiomyocytes don't proliferate, so we don't replace it. So the patched heart slowly starts to get bigger, right? It's like if you had 100 cardiomyocytes to do the job, now you only have 60 cardiomyocytes to the job. So 60 cardiomyocytes are now doing a job of 100 cardiomyocytes. So there is a lot of load on those cardiomyocytes, and the heart keeps getting bigger to accommodate that. That's a classical state of heart failure. So now what happens to immune cells during heart failure? And if you look at the spleen that way, again, during heart failure, spleen actually becomes bigger. It's an opposite thing that happens. And it is growing bigger because immune cells are proliferating. They are dividing in the spleen. They are becoming more and more immune cells, and those immune cells are going where? They are going to the heart, right? So immune cells are going to the heart. So if immune cells are going to the heart and they are protective, they are healing the heart, then we should see a protective response. Why heart keeps getting bigger, right? What is going on with the heart when it comes to immune cells? So in heart attack, you see a signal goes to the spleen. that spleen then shrinks, it becomes smaller because immune cells leave the spleen and go to the heart. What happens during heart failure is heart grows bigger, right? Again, a signal is still going to the spleen, but that signal is not reducing the size of the spleen, it's actually making the spleen bigger, right? And it's making the spleen bigger, these immune cells are proliferating, and those dividing immune cells are still going to the heart, right? Although we see immune cells going to the heart in both scenarios, but what is going on in the spleen is completely different. In the first one, immune cells are leaving. In the second one, immune cells are also dividing now, right, within the spleen. So what happens is, and what I call is like a frustrated response, right? Think about ourselves, right? We are all humans, and we try to do a thing, right? We try to do a job. For example, you're trying to understand a complex thing in your book, and you keep on trying, but you're not able to understand it, right? You're trying to do something, you're not able to do it, even after several attempts. T cells are doing the same thing. T cells try to heal the heart. They try, heart doesn't get better. If things don't get better with us, what happens to us, right? We get frustrated, right? And sometimes we just throw away everything, we leave everything and then go away, right? Or we get frustrated, we get angry, and then we do more damage, right, to whatever we are doing. Same thing happens with T cells. They get frustrated, right? They're trying to heal, so they just keep shooting those cytokines, chemokines around, and rather than helping the heart, those cytokines, chemokines start doing more damage because it's a different thing that's going on in the heart. Cardiomyocytes are dying, but not at that level now, right? They're slowly dying because there's a lot of stress. They're not dying because there is no oxygen now, right? So, but heart, T cells don't understand that, right? So this is what we do. We're trying to identify what goes wrong in these T cells, right? What is the signal that turns them from good during heart attack to bad during heart failure? And our science is looking to answer those questions, right? How can we prevent this transitioning of T cells from being protective to bad in our lab, right? So what you can do is you can give these T cells something, right? Something that can tone them down, that can calm them down, right? Say if you're frustrated because you're not able to grasp a concept, what happens, right? Your mother comes and says, oh, don't worry, beta. Things will be better, go take rest or go walk around, go talk to your friends, do something, right? Just calm down, right? So what we realized is that when these T cells are transitioning from good to bad, they activate certain proteins, right? And those proteins can be utilized, and those activated proteins are actually, we can target them, we can stop those proteins from functioning so that we can stop that frustrated response, right? I call that protein ERBA, and I'll tell you why we call it ERBA. This ERBA has the ability to calm down these frustrated T cells, right? It makes them stop producing all those cytokines, chemokines, so that T cells feel better again, right? Either they don't let them become frustrated, right? So that ERBA is estrogen receptor beta agonist, ERBA, estrogen receptor beta agonist, right? So we identified that these T cells, when they go bad, one of the receptors that's bad in them is estrogen receptor alpha, and we can stop that activation by activating other protein, ER beta. So it's a drug that actually goes and binds with ER beta and then makes it go to the nucleus and stop T cells from becoming bad, right? And when ERBA works, then T cells don't produce those cytokines, they don't produce those chemokines, and we think that they feel better. We stop that frustrated response from the T cells, and everything goes back, or everything at least happy the way it is. So heart doesn't become bigger after that, and heart stays wherever it was. We are not able to, we cannot reverse the damage, but we are at least able to stop further damage that these T cells are causing to the heart. So this is our work with T cells, and our design of this drug was actually featured in Time magazine last year, just to show that this treatment that we are working on, we think at least it has the potential to become the first drug that to stop heart failure progression in heart failure patients. So, So to acknowledge, most of this work was done by Rachel in my lab, Vinay and Austin, and Sydney Worth was an undergraduate student with me, and she's the one who worked on that comic book that I showed you some pages from, and we are looking to publish that for students, high school students, to read and understand the role of T cells and immune system in heart failure. Although I put this cartoon here, but I promise you I will not evade your questions, and I'll try to answer them as much as I can. With that, thank you for your attention, and I'm happy to take your questions now.
So we have time for 2 questions. Any volunteers?
So your speech is very inspiring, and I also want to be a teacher like you, sir.
Thank you. I'm glad. If I can persuade even 10 of you to become a teacher, I think my job is well done here today.
It's a very nice presentation, sir. And my question is estrogen receptor beta is responsible for producing chemokines in T cells, sir?
So T cells, that's a good question. I don't think it's responsible for producing them. They get produced because of other receptors, TCRs, right? Estrogen receptor beta is actually a transcription factor. Okay. So transcription factors, what they do, they get activated in cytoplasm, right? And then they go to the nucleus and start the gene program, right? Dr. McKinsey showed you how genes get activated, right, from that chromatin. So estrogen receptor alpha and beta are transcription factors. They activate those gene programs. Okay. What I think what happens is estrogen receptor beta goes and sits on those genes, right? Then whenever TCR mediated T cell activation takes place, there is already somebody sitting, right? Then other things cannot activate those receptors. It's like if you have one bathroom, right? Someone is already in the bathroom, the bad person cannot go in the bathroom, right? If somebody is doing that damage, right? It cannot go there. So you block access for that bad person to go in the bathroom, right? And then do more damage.
Thank you, sir.
Thank you very much.
Say it again, sorry?
What are the autoimmune diseases that affect heart?
So there are several of them, right? So autoimmune diseases develop against specific cells. Let me put it this way. And you can have autoimmune disease of pancreas, that's type 1 diabetes, right? You can have an autoimmune disease of intestine, that's an IBD, inflammatory bowel disease. And every tissue has the ability to have an autoimmune disease. So whenever a T cell gets activated against that cell of that tissue, it destroys that tissue, and that's an autoimmune disease, right? So some of those activated T cells, they can even go infiltrate the heart and lead to myocarditis slowly. But autoimmune diseases, it's an indirect response of activated T cells, I think. There are other myocarditis where you can have direct activation of autoimmune T cells as well, right? So we studied about myopathies in the morning. There are certain myopathies where you can have activation of T cells against myosin binding protein, and those T cells can go and infiltrate the heart. So that's an autoimmune disease of the heart directly.
Yeah, hi. Hi, I'm Dr. Anand. I'm a cardiac surgeon. I am really spellbound to see such a crowd talking hardcore cardiac sciences. My question to you is that at what level have you progressed with this ERBA? Have you gone to clinical trials, or what stage are you in?
Not yet. So what we have finished almost all studies in rodents, in mice. So we have safety data in them, toxicity data in them, and efficacy data. So we've done different doses as well. Even at 10 milligram per kg dose oral, we have excellent efficacy from this drug. We are looking to do pig studies now. Problem is it's a completely new drug. Nobody has it. So even to get it synthesized is almost $150,000. It's like pursuing opportunities to do that.
Thank you very much. They just communicated me that there's time for one more question. So if there's one more question, I saw one in the audience.
So whether we can use cardiovascular markers for to detect the immune system levels or increase in the enzyme systems to detect the T cells, T lymphocyte cells?
That's a great question. Cardiac markers are activated, are released from several reasons, right? Because of cardiomyocyte death as well, right? But I'll take your question in a different way. Maybe we can rephrase it in a way, can we look at T cells as a biomarker for heart failure or progression in heart failure, right? So what we can do is, in human patients, we have access to blood, right? So what we have seen, these T cells, when they get activated, they have higher expression of TNF-alpha. That's one of the bad cytokines and chemokines that we were talking about, at least during heart failure stage, right? So we can see how many of those T cells are producing TNF-alpha and know whether they are getting into that stage of frustrated response. Other way to do it is, maybe we can also look at estrogen receptor signaling, right? Estrogen receptor proteins, beta and alpha, and find out when it's increased, so that to start using this drug in them. So there are multiple other ways that we can utilize this as a strategy for biomarker and fine-tune our treatment for every patient, right? We don't have to give this drug to every heart failure patient at the same time, right? We can see when their T cells get activated by TNF-alpha expression, or when they have more estrogen receptor alpha or beta in them, and then fine-tune, individualize, precise, and have precision medicine that way, according to that. Yeah. Thank you.
Thank you, Shyam.
We express our heartfelt thanks to Dr. Shyam Bansal for his insightful session. Now, I request Dr. K.M. Shaktivel, Assistant Professor from the Department of Biochemistry, to felicitate Dr. Bansal.
Thank you, sir. We are privileged to introduce the notable speaker for session 7, Dr. Hossein Ardahelli. Dr. Hossein Ardahelli is the Director of the Center for Molecular Cardiology and Director of the Medical Sciences Training Program. As a Specialist Cardiology Fellow and Internal Medicine at the John awarded by NIH, John Hopkins, American Cancer Society, SQUEP Foundation, American Heart Association, and Chicago Biomedical Consortium. It is our honor to welcome Dr. Hossein Ardahelli to share his expertise on the topic, Role of Iron in Cardiovascular Disease. We kindly request Dr. Hindlal and Dr. Timothy McKenzie to chair and moderate the session.
Thank you very much. It's our pleasure to moderate the next two talks. Our first speaker was introduced very nicely. I just want to point out that not only is Dr. Ardahelli a world-class scientist, but he also takes care of patients with heart failure and heart disease. So unlike many of us who study heart disease in mice and rats, Hossein does that, but he also goes to the clinic to take care of patients. And that really gives him a unique perspective on what we're doing here. So thank you, Hossein. We're looking forward to your presentation.
All right, can you hear me? Can you hear me? Great. So thank you, Tim. I want to also thank the organizers for giving me the opportunity to be here. I've really enjoyed my visit to India. It's a wonderful country, and it has been an amazing experience. So thank you, everyone, for this opportunity. So I'm going to stand here. I don't want to block the screen, so you may see me standing over here for the whole talk. What I would like to do is that, if you remember, Dr. McKenzie told you that he comes from a background where he was very lucky. He had a lot going on for him. I want to show you that actually wasn't the case for me. I had a lot of barriers in my life that I had to overcome. And I hope that I give you the message, especially to the young crowd here, that anything you can overcome, and you can, you hopefully your ambitions will be achieved. So with that, I want to, okay. Okay, there we go. So I want to start with my dad, his immigration to the US. I was born in Iran. So this is the country that is, sorry, this thing doesn't, there we go. So this is a country here in the Middle East, close to India. Right, so unfortunately I can't use a pointer. So my dad in 1948, he went from Iran, he took an airplane from Iran to Beirut. At that time there were no direct flights to the US. And then he took a ship from Beirut all the way to the United States. And he went through Ellis Island. So there is, the Statue of Liberty, there is Ellis Island. So he went through Ellis Island, he went to the US. And he has told us a lot of really interesting stories when he went to the US. He went to the traffic light or an intersection, and he stood there for a long time looking at the cars, trying to figure out why cars stop all of a sudden at this intersection. And he couldn't figure it out. At the end, he thought there was a magnet in the ground that would stop the cars. There were no traffic lights in Iran, and it didn't occur to him to look up and see that there is a light that changes colors. So that shows you in 1948, he was very brave to make this big trip to the US. All right. So that was my dad, and this is actually what we found on the internet. You can, these records are available. This is from Ellis Island. This is his trip. He was on this ship, and the date is incorrect. It was in 1948, and he went to the US in 1948. So 3 years after World War II ended. But he actually went back. He went back to Iran after he got his PhD from Clark University. And then in 1953, he went back and he worked for the Point 4 program, which I believe Point 4 program also applied to India. The idea here was to prevent communism expansion to countries that are susceptible to get communism. So he worked for Point 4 program at the time. And I was the 6th child out of a 7-children family. So this was, I was in a large family, and I was born in Iran. So that's why, I was not really born in a country where I'm residing right now. And if I talk about Iran, these are mostly the images that come to mind, angry crowd burning American flags and hostility towards the US. But it's actually a very nice country, a lot of natural resources. I have, over the past 35 years since I left, or close to 40 years since I left, I've been there maybe 5 times. So I really haven't visited the country, but I would love to spend more time to go back and forth. And one thing I see from my colleagues, who are Indian or Japanese or Chinese, they can freely go back and forth, but I can't. So there is a lot of, there are a lot of things that I had to endure because of the politics of Iran. And again, this country has a lot to offer, and I want to show you, it has mountains. This is actually the city of Tehran in the close to the mountains. Where I grew up was actually on the bottom lower left-hand corner of that picture. So I grew up close to the mountains in Iran, and that's where I spent most of my childhood. Now, I want to show you this picture. In this picture, I don't know if you can identify who it is, but that boy sitting there. I was 6 years old, and the guy who is sitting next to me on the left-hand side is or my left hand, actually everybody's left hand. That's my dad. The girl behind me is my sister, and the guy is my uncle. Unfortunately, I'm the only one in that picture who is still alive. And that's what I want to start by telling you, the crisis and the crisis that I encountered in my life. This picture, I remember that night very vividly. My oldest sister, the next day after this night, immigrated to the US. So my dad had this philosophy that all of us, before we finish high school, we should go to the US. And my sister was in her last, or in the 11th grade of high school, and the day after this night, she flew to the US. So I was a child wondering, how can someone leave everything behind and go to another country and start everything all over? But the other thing I want to mention is that a couple of years after this picture was taken, a lot of things happened that were outside of my control, but had a lot of big impact on my life. The first thing was that my sister back there, She died in a car accident a couple of years later. And as you can imagine, I was very close to her. So that had a really significant impact on me. And it was a very traumatic event in my life. But if that wasn't enough, the Iranian Revolution happened. Many of you guys are young and you don't remember Iranian Revolution. I was only, I believe, 7 or 8 years old when it happened. But it was a really traumatic event, not just for the people of Iran, but for almost every country in the world. We had a very, powerful monarchy, but it was replaced by Khomeini, who was very oppressive, and his policies were to oppress anyone who did not have, who had any close relationship with the US. So this was a very traumatic event in my life. And then if that wasn't enough, so I already told you too, the other thing that happened right after this was the overtake of the American embassy in Iran. So that happened on November 4th, 1979. The embassy was actually close to our house. And I remember the day that it was overtaken. Very barbaric act, right? So for 444 days, the ambassador and also everybody who was working in the embassy, they were taken hostage. A very barbaric act that has never happened probably in the history of mankind. So because of that, the Iranian relationship with a lot of countries changed. Now, if that wasn't enough, then the next thing that happened was the war between Iran and Iraq. And that lasted about 8 years. And at that time, I was young enough to be automatically registered in the military service. So everybody who started high school was automatically registered in the military service, and we were not allowed to leave the country. They needed young boys to go to the war. So I couldn't leave the country. And I want to show you a couple of pictures from that war. I could be this guy who is standing here. There were young kids who were given plastic keys, and it said made in China. And they were told that these are keys to heaven. And they were asked to run on minefields and sacrifice their lives to open up the minefields for the soldiers to go and attack the Iraqi soldiers. And I could have been one of these kids if I wasn't fortunate enough for my parents to have the resources to get me out of the country. I'm not going to go over the details of how I got out of the country. I really hope one day I can write a book on that, because it was a very, very, very complicated way to get out of the country when I'm supposed to serve in the military. So anyways, I got out of Iran, and many of these pictures you're seeing, they look like pictures from World War I, right? So they have these trenches where soldiers were, chemical weapons were used. So almost everything that happened during that war was a duplicate of what happened in World War I, and a lot of young, innocent people were killed during this war. So with that, again, I left Iran in 1985. I was, at that time, 15 years old during high school. We were in Europe for 4 months, and we entered the US in February of 1986. And I finished my high school in Fremont, California. And then after that, I got my bachelor's degree actually in computer science from the University of Utah. And then after that, I went to Vanderbilt. Actually, Tim McKenzie and I, we graduated in the same year, 1998. But we never met. So we actually had the conversation, I believe, in Hawaii, and we realized we were actually classmates at Vanderbilt. So I got both my MD and PhD at Vanderbilt. And I did my residency and cardiology fellowship at Johns Hopkins. I'm just going to make a joke here. It's actually Johns Hopkins. A lot of people think that it's John Hopkins. So there was one time the chair of medicine at Johns Hopkins was invited to give a presentation in Pittsburgh, and he was introduced to be from Johns Hopkins. So he got up and he says, "It's great to be here in Pittsburgh. So please don't forget the S in Johns Hopkins. That's very important." All right, so current position, I'm actually a professor of cardiac metabolism, professor of medicine, cardiology, and pharmacology. I'm very honored that I have served as the president of American Society for Clinical Investigation, which is the honor society for physician scientists. You have to be accomplished to get into it. 2 years ago, I was humbled to serve as the president, and right now I serve as the deputy editor of the Journal of Clinical Investigation, which is believed to be the most prestigious journal for physician scientists and for papers that are mostly mechanistic studies. But what I've accomplished a lot, but what I am most proud of is that I have a wonderful family. I wanted to show pictures of my son and my daughter, who my daughter is finishing medical school next month. So I'm really excited that she's, and I'm going to be actually hooting her at the graduation. So that's basically a summary of my background. And I now want to tell you about the research we are doing in our lab. There are two major projects we are doing. One of them is on hexokinases. And when I was at Vanderbilt working on my PhD, I worked on this enzyme. How many of you guys know what hexokinases are? By show of hands. All right, well, very few, and I'm actually surprised. So hexokinases are enzymes that phosphorylate glucose as glucose gets into the cell. So basically, it traps glucose inside the cell. These are very important enzymes. If you don't have hexokinases, you cannot metabolize glucose. Why am I interested in these enzymes? Because mammalian hexokinases have evolved to become very complicated. If you just need to phosphorylate glucose, you just need one of those red bars. But they have become very complicated, and we have shown in mouse models that each portion of these enzymes, as they evolve, they have gained additional functions that regulate glucose metabolism in general. So this has turned into a very interesting project. Today I'm not going to talk about this I'm not going to talk about this project, but what I want to talk about is iron in cardiovascular disease. And I want to show this slide. I got this slide from my friend Joe Hill, who is the editor of Circulation. And I just want to emphasize, in the field of iron in cardiovascular disease, we always say that there is disconnect between basic science and clinical science. I don't think there is any field in cardiology where there is a bigger disconnect between basic science and clinical science than there is in the field of iron in cardiovascular disease. And that's why I want to really focus on that today briefly and tell you what we are doing on that. I'm going to tell you a lot of clinical studies that have been done on iron in cardiovascular disease, and I think it's relevant. I see a lot of young women in this room, and many of you are probably iron deficient, especially in India, because this major source of iron in our body is meat. Heme is the best way to absorb iron, so iron deficiency is actually very common in the world. And in fact, the most common nutrient deficiency in the world is iron deficiency. And we see it a lot again in India and in sub-Saharan Africa. So I think this will be very important for the audience here. So let me tell you first about heart failure. You have heard this a lot before, but heart failure this is the normal heart, but there are two forms of heart failure. One of them is what you have already heard. It's called heart failure with reduced ejection fraction, and that's when the pumping function of the heart is reduced. Now, everybody's, you learn this in college, that heart is a pump, right? That's what you have heard. That's half of the story. The heart is a pump, but it's also a sucking device. It has to relax to allow blood to get into it. So if the pumping function is reduced, then you get HFrEF, or heart failure with reduced ejection fraction. When the elasticity, when the relaxation of the heart is affected, then you get HFpEF. Now, this is, HFpEF, that's just half of this story. And a lot of us focus on the heart as the major organ to be involved in HFpEF. But probably the majority of the defect in our body in HFpEF is actually in our blood vessels. Our blood vessels lose their elasticity. And that's a problem in the field of cardiovascular research, that we are all focusing on the heart. And that's probably actually just not a big part of the disease. It's most of our, the problem is in the vessels. What is the cause of heart failure? I intentionally put this slide, I wanted to show this to you, because this is not accurate. This is probably what was the cause of heart failure, 20 years ago. Coronary artery disease. Now it's a lot of other things. It's high blood pressure, diabetes. As we live longer, most of us are going to get HFpEF, or heart failure with preserved ejection fraction. In fact, HFpEF, I would consider HFpEF not a disease. I would consider it as a marker of aging. I bet you if we live long enough, all of us in this room, if we live long enough, we all will get HFpEF. It's a marker of aging. Some of us get it earlier because we have comorbid conditions. Some of us don't. So if you consider aging as a disease, which probably is, then HFpEF would also be a disease. But it's not really a disease on its own, and it's just a marker of how somebody has aged because of their risk factors. So what are the symptoms? These are the symptoms of heart failure. You have low exercise capacity. You have shortness of breath. You cannot lie flat on your back. I ask my patients, how many pillows do you use at home? Sometimes you have to go and go to the bathroom a lot. You have swelling and weight gain. And then the signs, I'm not going to go over this, but these are the things we use to examine our patients and make the diagnosis. There are different classes. And the higher the class of heart failure, the worse it is. I ask my patients, can you walk to your mailbox to get your mail? And some of them say, no, I get short of breath before I get to the mailbox. And that's basically New York Heart Association class 4. They have severe limitations and they have symptoms addressed. And the higher your class, the higher your mortality. So our goal is to make sure that we don't get to that level. So that's basically a summary of heart failure. And again, what happens is that these patients go home. They come back because there was a precipitating factor. What was the precipitating factor? Most of the time is diet. In the US, diet, for older people is microwave dinner. They just put it in the microwave and they eat it. It's very high in salt. And that causes them, to come back to clinic. And this continues until they get into complete decompensation. So, how many more minutes? Okay, so he said I have 30 minutes. Anyway, so I want to talk about IV iron in cardiovascular disease. So I'm going to go over this really quick. But basically, it's becoming a billion-dollar industry. IV iron, which means giving iron by intravenous mode, has become a major mode of therapy in patients with cardiovascular disease. These are the recommendations by the American Heart Association and also by the European Society of Cardiology. This was valid until last August. Last August, the European Society for Cardiology changed their indications, and they gave it a class 1 indication. What does it mean, class 1 indication? That means everybody who comes to the hospital with heart failure should get IV iron to improve their symptoms, which is basically not based on science. So let me tell you why. There have been a lot of studies that have been done here. So we have published, these are all the papers we have published in a lot of journals arguing that we are moving too fast. Americans are a little more careful. Europeans have moved really fast, and they have put this into their guidelines. So what are the differences of iron? What are the different types of iron deficiency? There is absolute iron deficiency. If you're iron deficient and you're in this room, you probably have absolute iron deficiency. You don't have enough iron in your body. But there are people who have chronic inflammation, patients with cancer, patients with autoimmune disease, they get functional iron deficiency. And that's because they have enough iron in their body, but it's basically trapped in their macrophages. It's not available for them to use. So it's called functional iron deficiency. The reason is because of inflammation, this hormone hepcidin is increased, so they do not absorb from the intestinal tract, and they do not release iron from their macrophages. And what are the symptoms? Again, you have, they have coarse, lining on their tongues. They have, the changes of their nail, and they lose hair. So that happens in these patients. So the problem is that in patients with heart failure, iron deficiency is defined with ferritin. Sorry, this thing is not moving fast. It's ferritin cutoff of 100 nanogram per mil. If it's defined as iron deficiency, and if your ferritin is between 100 to 300, TSAT of less than 20. I bet you almost all of us in this room will have our ferritin less than 300. So if we get heart failure and we go to the hospital in Europe, they're going to give us IV iron. That shows you how loose these criteria are to give IV iron to these patients. And the reason is because, of course, you have to create a disease if you want to sell your drug. So the criteria has been defined as very loose for this condition. Now, what do we know about iron and cardiovascular disease? I'm not going to go over all of this in great detail, but one thing I want to tell you, that blood donors have lower incidence of cardiovascular events if you correct for everything else. And that's why I try to donate blood on a regular basis. In the US, you cannot give it more frequently than every 3 months. So I try to give blood every 3 months. It's good for healthcare in the US, and also it's good for that. Now, I want to show you a few studies, clinical studies. I highlighted what needs to be done. I'll be done in a second. Basically, all of these studies did not meet their primary endpoints, which was to show there was a reduction in cardiovascular events. Ironman didn't show it. AFFIRM-AHF didn't show it. And this study, if there are 2 curves that can be superimposed any better than this, you cannot find them. So there was no evidence that IV iron helps in patients with heart failure. So despite this, a lot of people are given IV iron. And the FDA label is also in a way that patients who they have to try oral iron first, and also in patients with chronic kidney disease. Okay, so how about oral iron? Can it be used in patients? Yes, there is. But there was this study where they used oral iron versus placebo, and they didn't see any improvement. So they basically wanted to use that. A lot of clinicians use that to say that oral iron doesn't work in patients with heart failure. All right. I'm going to, if you can give me 2-3 minutes, because, so TAC trials basically uses chelators, and they showed that there is benefit in patients with coronary artery disease. There was 41% reduction in the risk of any cardiovascular events in these patients. And IV iron has major side effects. I'm not going to go over all this in great detail, but osteomalacia is one of them, and infection. Infection is very common in patients with heart failure. In fact, there was a paper that was published recently that showed that infection is very high. We are doing a lot of studies. Now we are doing clinical study on the role of iron chelators in patients with coronary disease with these 2 investigators in LA. So over the past few years, we have published a number of papers on iron in cardiovascular disease. And I'm not going to go over in great detail here, but basically we showed that if there is a reduction in mitochondrial iron, it's protective against cardiovascular events. And we identified cellular iron to be regulated by this pathway called mTOR and TTP. Let me skip over all of that, but that's basically a summary of what I've already told you. I'm going to skip that, and I'm going to go to the last slide here. So, and again, most of this was done by really smart MD PhD students and postdocs in the lab. All right, so this is my lab. I want to thank them. Before I end, I want to also show you this slide. How many of you know this poet on the next slide? How many of you know Rumi? Anybody? So I thought more people in India would know Rumi. Rumi is basically a Turkish Persian poet, and he basically started Sufism. And he had this poem that I thought was a very nice poem that basically defines people who overcome a lot of barriers in their life. He says, I was dead and became alive. I was tears and I became laughter. The reign of love began and I became eternal. For those of us who are doing research, if we overcome our barriers, our impact on science is eternal. It's not because of the science we are doing, but also the training we are doing for our trainees. And they can go on and train more people, and this can continue and duplicate. And finally, I want to mention that we also have JCI at Northwestern, and it's a mechanistic journal. And if any of you are interested, we would be happy to look at your papers. Thank you very much, and I would be happy to answer any questions. So it was 25 minutes.
Thanks, Hossein, for a beautiful talk. We have time for maybe 1 question or 2 questions. Anyone?
Yes.
Sir, I have a doubt, sir.
Yeah.
Sir, I'm here.
Go ahead.
Sir, here. MC desk.
Yeah.
I I just have a small clarification regarding this iron deficiency.
Yeah.
If iron deficiency is happening because of the vitamin B12 deficiency, which is caused by the autoimmune disease,
Right.
is the mecobalamin only way, which is in the form of injection or the tablets? What I would know is that can we reverse this vitamin B12 deficiency which is caused because of the autoimmune?
Right, so vitamin B12 deficiency can happen because, vegetarians don't absorb vitamin B12. It has to be in the form of, meat, or it can be because of pernicious anemia. Pernicious anemia is when you have autoantibodies against your cells in the digestive tract to absorb B12. So you need to get the injection, right? So if you don't get B12, then that's a different kind of anemia that you get. It's called macrocytic anemia. But with iron deficiency, you get microcytic anemia, and the forms are completely different. The cells, or red blood cells, become hypochromic, and they're smaller with iron deficiency. With B12 deficiency, the cells are bigger, and the red blood cells are bigger. So they're completely different pathways. There was one question here. Yes, sir.
Sir, yes. Sir, if iron is given in intravenous condition, won't it cause hemosiderosis? Accumulation of iron causing problem in liver?
Right, so no, actually that has been, the idea here is that when you give IV iron, iron is actually absorbed by the macrophage and it's released into the bloodstream. So usually it doesn't cause it. However, there is a paper that hopefully will come out soon, not by my group, that they have shown that iron does accumulate in the heart if it is given by IV iron. Excuse me? Right, so I just told you that there are side effects. So it can increase the risk of infection, right? We have a system in our body to prevent infection or fight infection by withholding iron from bacteria. And that's why hepcidin is released. Hepcidin is actually bactericidal by withholding iron from bacteria. So if you give IV iron to those patients, the risk of infection goes up. There is, with ferric carboxy maltose, there is an increase of osteomalacia that can happen. The bones can become weak. And there is the risk of course, coronary artery disease that can be increased. So those are all risk factors. Okay. Thank you, everyone.
Thank you, sir. Now I invite Dr. M. Sivaselvakumar, Associate Professor, CMMT, PSGIMS and R, to felicitate Dr. Adehelli.
Thank you, dignitaries. We are privileged to introduce the erudite speaker for session 8, Dr. Satyamangala V. Nagaprasad. Dr. Satyamangala is the Professor of Molecular Medicine and Department of Cardiovascular and Metabolic Sciences at Case Western Reserve University. He completed his BSc in Osmania University and PhD at Jawaharlal Nehru University, India, followed by postdoctoral training at University of North Carolina and Duke University. Since 2005, in Cleveland University, as an independent faculty, he has established an overarching program that encompasses receptor pharmacology, biochemistry, genetic engineering tools, and cardiovascular physiology. He discovered the association between PI3K gamma and protein phosphate through several collaborative researches. He has received research grants from NIH and AHA with several breakthrough research outcomes published in reputed journals like Nature Cell Biology, Circulation, and Molecular Cell. He has 75 plus publications in high impact journals like Scientific Reports and Circulation, and has been honored with 19 plus awards, including the Science Research Prize for Young Investigators, winner of the Dean's Prize for Medical Student Research. It's my honor to welcome Satyamangala V. Nagaprasad to share his expertise on the topic Novel Pathways Regulating G-protein Coupled Receptor Function in Heart Failure. We kindly request Dr. Hindlal and Dr. Timothy McKenzie to moderate the session.
Hello. So Dr. Prasad is a world-renowned scientist for working on the G protein coupled receptor signaling. So in addition to a great scientist, I just like to add a small note that he's role model for many, certainly for me, because of his simple personality and straightforward while working. So he is like mentors for many. With this, let's welcome Dr. Prasad.
Can you all hear me? Yeah, okay, thank you. So, thanks very much for the introduction. I thought I'll put the slide up. You can see in my name Satyamangala, but actually it's Satyamangalam. So my ancestors were from Satyamangalam, and that's my grandmother who is from Gopichettipalayam. It's not very far away from here. So my grandfather is from Satyamangalam, and my grandmother is from Gopichettipalayam. But I had a story to tell because my grandfather married her when they were very young. So my grandfather moved to Hyderabad where as a Tamilian he didn't know anything, but he learned Urdu and became a professor of Urdu in University of Osmania. But when they were married, they were at different heights. But after that, they found out after 10 years that my grandmother was taller than him. And so that used to be why we don't have a picture together. But my grandmother passed away a long time back. So that's the story behind my name Satyamangalam. And I think, yeah, this is where my story begins. So I was born in Chennai, did my schooling in Hyderabad, and then from there, I went to do my PhD at Jawaharlal University, went for postdoctoral training at Duke University, and that's where I think I got great amount of mentorship. And my mentors, one of them is a Nobel Prize winner in 2013, Bob Lafkowitz. And I work with G protein coupled receptors, and that's where my lab works on. After that, I moved to Cleveland Clinic to become an assistant professor. But there's one more side story that I think you all should learn from this. So if your father is very adamant, you have to give it up, okay? So that's what you can do. So I actually wanted to be in the Army. So when I was in 10th grade, I actually applied for NDA certification, but my father wouldn't sign it. So then I said, okay, let me become a doctor. So I wrote the exam. I got a seat. But the seat was not in my city. It was in the Naxalite-prone city of Warangal. And my father said that I'm not going to sign it. So then I actually joined a BSc program. I failed in my first year BSc. Still my father wouldn't budge. So then I said, okay, now I need to turn my life around. And then that's how I turned around the life. So it's not that what you lose, but I think what you look forward to makes you where you go. So it's not that, oh, I can't do that, this did not happen. It's not where you think life. You say, okay, this is what I got now, and how best I can make use of it. So that's where I think it's a learning lesson for everybody, that you always have to be positive in your life. Some things happen for good, some things happen not for good. But I think this happened for good because otherwise I wouldn't be standing here with a great set of people talking about this. I would have been a clinician practicing somewhere in Hyderabad. Okay, so this is where my lab works on, and I got a lot of science today rather than all the other people who talked about, because I'm going to talk about something very different, very fundamental of what we're going to do, and it's got its implications in cardiac outcomes. Anita in my lab is the one who did this project. She's a very brave soul, because you can see how difficult this work is going to be, because we didn't know the answers for it, and we don't have answers for it. So I'm presenting this because this shows you that actually you can be open about what you're thinking, and that openness gives you an ability to think outside the box, okay? So I'll tell you our journey through this process and how it opened up new ideas for which you don't have answers, but at least we can prove what we're looking at. So the topic that she told was wrong because I thought I'll present something that is very different, and today it's going to be unraveling the role of mitochondria. So classically, all of you know what's the mitochondria. Mitochondria is a powerhouse of a cell, right? All of you think about it that way. But we think differently. And that's why I said Anita is a brave soul, because hopefully that work that she did can convince you that mitochondria is not just a powerhouse of a cell, but it's got new roles to play, and that role is critical for fibrotic changes in the heart. So you heard all my esteemed speaker talk about fibrosis, and I'm going to put that information of how this fibrosis happens and how mitochondria could be a key part in this process. Okay, So everybody talked about heart. Heart is a 4-chamber organ, and that's how a normal heart looks like. Hopefully it changes. Okay. And this is also an image that all of you saw, where whenever there is less loss of oxygen in a different part of the heart muscles die. And as the heart muscle cardiomyocytes die, they are replaced by fibroblasts, like Dr. Lal, Dr. Timothy McKenzie talked about, and also Jonathan. And then when you look at it, the heart has to be an elastic organ, right? And in that process, it's replaced by these fibroblasts, which get activated. And that activation leads to remodeling. That's what leads to what is called a dilated cardiomyopathy. So in our lab, we use mouse models. And the reason why you use mouse models is you can actually follow them non-invasively. Shown is a simple echocardiography of a mouse from the left to the right, where you can see that when you take a normal mice and put them into stress, this mice undergoes cardiac remodeling, and that remodeling can be measured by the difference between the two white lines that you see. When it's the shorter white line, that's called a systolic curve, and the one that's larger is diastolic. The difference in measurement is what we call fractional shortening or ejection fraction that you measure at. Okay? Okay. So we work on this enzyme called PI3 kinase. Hopefully you all know. How many of you know PI3 kinase by any chance? Oh, wow. That's good. I thought we nobody. But yeah, so this is an important enzyme because this enzyme is absent in normal tissues, but gets activated when an organ gets stressed. So that's why it's a very important enzyme for us. And this is a description of how it works. And so if people who want to actually look at in-depth in terms of signaling pathway, PI3 kinase gets activated, and then it plays a role in actually being an anti-apoptotic process. And that's why you get an upregulation of this enzyme when there is cardiac stress. So what we did long time ago, when I was a fellow, was to actually make a mice to inactivate that and see what happens. And when you subject this mice, to heart failure mouse model, you can see on the top panel, by 12 weeks of heart stress, you can see a dilation in the heart that is still maintained in inactive PI3K, suggesting that if you just target this particular protein in the heart, you can actually get a rescue in function. So we went back and then said, okay, what happens if you actually take away the enzyme? And that's where the surprise comes. If you take away the enzyme, you can see that there is more fibrosis than the wild type. That means that when you actually injure a heart of a mice that doesn't have this small enzyme, you actually get more fibrosis. And so this surprised us, and we said, okay, we need to figure out what's happening, because that means that in these hearts, the fibroblast that's being talked about that replaces the elasticity of the heart is getting activated faster. And we use mouse models like what people talked about. We can actually tie that band around the aorta, and you can see actually the remodeling as measured by the staining. So since we have all these tools, we said, okay, let's go up and figure out why in this mice model such a thing happens. So this is basically a description to say that whenever there is a cardiac stress, you have these small cells called fibroblasts. They're very agile, and then they get activated and replace the actual injured myocardium. So this basically describes the overall phenotype that you would see. What happens are multiple layers that you all heard about, different people talking about different things, that actually ends in a fibroblast, that's a small one right on the top, becoming myofibroblast that is larger in size, that now replaces the dying myocyte. So the basic concept is that when you have a very small fibroblast, that's such a small cell, becoming such a large cell in such a short span of time, then how is that possible, right? And classically, it is known that the changing of this fibroblast that is so small to becoming this large cell involves factors that are sent out by dying cardiomyocytes. And the question is, what are those factors? And if so, how come a cell can become this such big cell in such a short span of time? So this transition normally takes probably 48 to 72 hours. And the question is, how does it happen? And this is where the challenge began for us, right? So if we went back and look at these cells in the heart from this mice that doesn't have this one single enzyme, you can see that these cells are forming myofibroblasts even at baseline. So the question is, why? So we went to find out we wanted to find out what these factors are. And so what we did was we actually took myocytes from these hearts. The only difference between the wild type and the knockout cells, absence of this one enzyme, and then looked at what proteins are being secreted out of the cell. Our idea is that if dying cardiomyocytes sends out signals to the fibroblasts to become these large cells, what are those factors? And what we surprisingly found was that 90% of the proteins were mitochondrial proteins. And so the question is, how come outside the cell there are so much proteins from mitochondria? That is a key question, and that's where our project began. And today, hopefully, I can convince you that mitochondria, that's supposed to be energy organelle of a cell, can come out of a cardiomyocyte, move from the cardiomyocyte into a fibroblast, act as a signal, and change the fibroblast phenotype from being a small cell to a large cell. Hope I can convince you, and then we'll see whether I can convince you or not. Okay, so that is our hypothesis. The idea is that when the cardiomyocyte becomes stressed, it sends out mitochondria, and that mitochondria now can change the small fibroblast to the big fibroblast. So the idea is that this is an organelle moving from one cell type to another cell type, and that cell type that receives this organelle changes the phenotype of that cell. Yeah. So people, all of you know about this, but I think the most important thing I would like to highlight is mitochondrial progeny is primarily contributed from the mother. So all of us carry mitochondrial DNA from the mother. That's the most important part. So mothers are very important. So what we did first was to actually show, we tested whether mitochondria comes out of the cell. And what we did was simple different kind of staining. Shown here is a staining for a dye that identifies whether mitochondria is existing in the supernatant or not. And you can see that there are more red dots. The red dots indicates that mitochondria is still active in the surface or secretome of the cells that we have actually plated. So this is the first proof of concept that when you actually plate these cells, let it settle, take the supernatant, and then stain for mitochondria, you can see mitochondria coming out of cardiomyocytes. So the next thing that we did was to do an electron scanning micrograph to provide proof of concept that cardiomyocytes can actually send out mitochondria. So shown here are the cardiomyocytes on your left at a lower magnification. And then when you move to the middle one, you can see that in the wild type as well as in the knockout. So there's only one enzyme missing in these two mice, or the hearts. And you can still see mitochondria being secreted out of the heart. So our idea is that cardiomyocytes can actually secrete a power organelle from one cell, sending it to the other cell to do the job. And this is to actually show that if you take the supernatant of the cells, which you plated, and do an electron micrograph, mitochondria being on the supernatant. So this is to show that if you actually have a cell that can secrete mitochondria, you can image it. That's a proof of concept to show that cells can secrete mitochondria, and that receiving cell can take this mitochondria and do the job that is required. The question that then we ask is whether it can be taken up by cells, right? So if one set of cells secrete it, doesn't mean other cells can take it. So what we did was we stained two different kinds of cells were stained with two different kinds of stains. The mitotracker green is a staining for the endogenous mitochondria in the cell. The H-rhodo red staining is the staining of the cell that we did. So the mitochondria that we got out of the cardiomyocytes, we stained and then plated onto these fibroblasts, and then we imaged them. And what you see on the right overlays that fibroblasts can actually take up this mitochondria that has come out of the cardiomyocyte, showing that the cells that are fibroblasts can receive this mitochondria, take it up, and then the question is, what do they do? Okay? So this is what happens. So I told you that cardiomyocytes secretes mitochondria. This mitochondria is taken up by cells, and our hypothesis is that they drive fibroblasts to a myofibroblast transition. Myofibroblasts are these large cells that are procontractile compared to fibroblasts that are very small. Okay? And that's what you see. If you compare, the staining here is for alpha smooth muscle actin. As Tim talked about it, he's used it for scanning. We just looked at the phenotype, and you can see that in the control there is very little scanning, very little alpha SMC on the left, if you take mitochondria either from the wild type cardiomyocyte supernatant or from the knockout supernatant and put them onto simple fibroblasts, you can see that from the small cells that you see on the left, they become large cells, showing that these fibroblasts can take mitochondria from these cells and become large. You don't need any other stimulus, right? On the right is a positive control to show that this experiment worked. So I've got 3 more slides, and so these are all in vitro kind of studies, right? So we actually wanted to now go back and say whether this happens in vivo. So what we've done is we've generated mice where we only have mitochondria in the cardiomyocyte that is green. You can see on the left, the top panel is a wild type myocyte that doesn't have any green mitochondria. Lower panel is a mice that has got mitochondria that's green. Shown here in the middle is a proof that it's a cardiomyocyte. And then we can see in the cardiomyocyte, the wild type doesn't express the green, but the lower one that expresses green mitochondria can be overlaid. Now you take these mice, right, and then you look at fibroblasts in these mice. Fibroblasts are already in the heart. So what we did was we just digested the heart and then looked whether the fibroblasts are green in color, because the mitochondria in the cardiomyocytes are green, and if there is a transfer endogenously from the cardiomyocyte to the fibroblast, the endogenous fibroblast should be green, and that's what you see. So what you see in the middle, on the left is the endogenous mitotracker red for all the endogenous fibroblast mitochondria. On the middle is the green mitochondria that is coming from the cardiomyocyte. It's not from the fibroblast, because only cardiomyocytes in this mice express the green mitochondria. And so this is a proof of concept that even in the mice, there is an active crosstalk between different cell types, in contrast to what is classically known that, oh, there are different hormones that occur between different cells. You can have now mitochondria moving from one cell to another cell, and that in turn can change the phenotype of the given cell system. And so, as a proof of concept, what we did was that we took basically the blood, purified the mitochondrial fraction, and then stained them. So, shown here in the bottom is the mitochondria from in the blood from these mice that only expresses mitochondria, green mitochondria in the myocyte. And what you can see in the circulation is that you can see green mitochondria present in the circulation from these mice. So this is first proof of concept that cardiomyocytes can secrete mitochondria out into the circulation, and that you can potentially use as a biomarker if you want, if you think about it that way. This is actually a more complex kind of a mouse model where every organelle in the mice is stained with red. It's called a mitocate mitochondria. And then we have green mitochondria only from the cardiomyocyte. So you're seeing endogenously that mitochondria, only green mitochondria from the cardiomyocyte getting into a red mitochondria of the fibroblast. So we have not done any changes from outside. It's all endogenous, showing that there's an active transport of an endogenous mitochondria from one cell to another cell type. So maybe this is too blurred, but the idea is to actually show what does mitochondria do. Mitochondria actually does electron transport, and electron transport leads to ATP. So our hypothesis is that when mitochondria is secreted by the cardiomyocyte, it's taken up by the neighboring fibroblast. Now that mitochondria in the fibroblast changes the phenotype by providing more ATP to this growing cell. So therefore, a small cell can become a large cell in a short span of time. And so to test this, we actually used what is called a mitochondrial assay. And uniqueness of the cardiomyocyte mitochondria is that it uses fatty acid compared to a normal cell mitochondria, which uses glucose. Okay? So that's how we can distinguish between a cardiomyocyte mitochondria and the mitochondria from other cells. So now shown here is a complex kind of a graph. You can just focus on the left. So what we have done is we have taken fibroblasts, treated them with mitochondria or no mitochondria. So we have three different bars there. One is green, one is red, one is blue. The blue bar on the left that you see is just normal fibroblasts. They have not received any mitochondria, and they have the normal pattern of respiration. As you see, the respiration goes up and comes down. But if you actually put mitochondria from either the wild type or the knockout mice, you can see that there are different levels The green one shows higher because this mitochondria are more active in the knockout. You're figuring out why. But you can still see that when you actually stimulate them, there's similar amount of energy that it can generate when you provide glucose as a substrate. But this is where the actual difference comes. And when you provide, instead of glucose, you provide fatty acid, which is selectively used only by the mitochondria from the cardiomyocyte, you can start seeing the difference. So the endogenous fibroblast that is shown in blue responds only to that much amount when you have fatty acids. But when you look at the fibroblasts that got mitochondria, both of them respond differently compared to the control. So this suggests, or is a proof of concept, that if you take mitochondria from one cell type and give it to another cell type, you can actually change the metabolic phenotype of a given cell. Also suggests that mitochondria could be a great signaling molecule between different cell types that leads to a different phenotype. I think, yes. So with that, I think I will just summarize our studies to show that mitochondria could move from one cell to another cell, a concept that we think is very interesting, very novel, compared to what is classically known, that mitochondria is an energy organelle for the cell. Second is that they actually carry their metabolic memory. So they carry the memory that they are in the cell and take it to the next cell and provide that memory to provide metabolic support. And finally, the idea is that if this metabolism happens, if mitochondria move from one cell type to another cell type, then they change the actual phenotype by changing metabolism. Why this is important is because in conditions of heart failure, you actually need to repair the given heart in a very short window, and that short window is only 42 to 72 hours. And so the small fibroblasts can become large myofibroblasts using the mitochondria from the cardiomyocytes to repair itself. So I think with that, I'll wrap it up, and I'm open for questions.
Thank you. We have time for a few questions.
Good evening, sir. Sir, I want to know what is the mechanism of mitochondrial uptake by the fibroblast cells, sir. Is there any way we can block the uptake of mitochondria?
Yes. So I did not share the study. So we are basically trying to say it is actually energy dependent mechanism. So there are markers on the cell surface of the fibroblast that can actually interact with the mitochondrial vacuole. It takes it up. So if you block it, then what happens is you won't get a transition from a fibroblast to a myofibroblast. And that may be deleterious for a given heart. I may be contrasting Tim, but idea is that you need a fibroblast transition.
Thank you, sir.
Oh, there are millions of mitochondria. Cardiomyocyte is one of the mitochondrial rich cell. And so when they release this mitochondria, either because of stress or because they die, your mitochondria coming out. And so that's how cardiomyocyte is a very rich mitochondrial
On the
cell.
average, how many mitochondria will be there in each cardiac muscle cell?
I don't know in average, it's in millions. I can tell you that.
Okay.
Yes.
Sir, is there any way we can change the myofibroblast back to normal fibroblasts?
Oh, that's a great question. So we believe that it is I think Tim can also join me in this answer. So we believe that once a fibroblast becomes a myofibroblast, it's already a universal, unidirectional transformation. And the idea is that once it becomes procontractile, it's very difficult for it to be reversed back into a fibroblast. And so the question is, if you want to make a myofibroblast that is beneficial for the heart, you want to have it permanent. You don't want to reverse it. Because what happens is the myocytes die. As myocytes die, you want them to take up the cardiomyocyte space so that the heart doesn't open up. And so it's normally a unidirectional process.
Thank you, sir.
If no more further questions, let's congratulate Dr. Prasad for a great talk.
Thank you.
Our heartfelt thanks to Dr. Satyamangala Nagaprasad for his enlightening session. Now, I invite Dr. Merlin Veronica to felicitate Dr. Prasad.
podium.
Thank you, madam. Thank you, sir. We are privileged to introduce the next speaker, Dr. Rajshekharan Namakal Surappan, Associate Professor from the Department of Pathology, University of Alabama. He completed his BSc at Bharathidasan University, MPhil and PhD in Biochemistry from University of Madras, and acquired postdoctoral training at IIT Madras and University of Utah. He is a visiting professor at PSG Center for Molecular Medicine and Therapeutics, PSG IMSR Coimbatore. With research focus on redox balance and antioxidants, he has been honored with 16+ awards such as the Paul Dudley International Science Award by the American Heart Association. He is the editorial board member in reputed journals, namely Frontiers in Oxidant Physiology, Cell Biology Research and Therapy, Reactive Oxygen Species, and Frontiers in Cell and Developmental Biology. As a reviewer for several biochemistry, physiology, medical, and cardiac-based journals, he has 80+ publications with more than 8,500 citations and an I-10 index of 58. It's an honor to welcome Dr. Rajshekharan to share his expertise on the topic, Does Redox Matter? A tale about the discovery of reductive stress.
அனைவருக்கும் வணக்கம். Good evening, everyone. It's my pleasure to be here, and I think I'm the last speaker because I'm not away from this institution because I've been collaborating with this institution for more than 6 years. So as said, today I'm going to talk about, everyone said about their stories from childhood. I don't have any slides for my own personal things, but I do have a strong story how the discovery of reductive stress has occurred, which is 17 years old. Still it is in its teenage, and I can walk you through where are we going, how we discovered, and what's going to be the future. Now I request you, all of you, to stand for 10 seconds to pay the tribute to our beloved former Dean of PSG IMSR Medical Institution, with whom I have collaborated. In fact, who is the brain behind the interest for collaborative research between the basic science and clinical science. And with his efforts, we were able to publish papers in collaboration with University of Alabama, Birmingham, as well as the PSG College. Thank you. Hopefully everyone is awake now. So here is my overview. Sorry. So I'm going to walk you through a little bit on the redox. Does redox matter? That's the first question I would like to discuss. And then a little bit about the redox spectrum. How do you look at redox? And the discovery of reductive stress in the mouse model or in the human disease of cardiac nature. And I'm going to show you an exclusive model for reductive stress in a mouse heart, and also discuss the little bit of signaling, which is nothing but the transcriptional aspect of redox, and then how reductive stress can influence the heart for pathological remodeling and dysfunction. How many of us, many of us know this. This is to students. Do you remember these guys? Do you know these guys? Did you read about them? They're nothing but Vijay, Surya, Ajit. Is that right? We know them, but not these guys, right? So these three individuals were awarded with Nobel Prize not too long ago, like in 2019, for their wonderful discovery. It is not a very high-fi molecular biology, but it's a very basic discovery showing that these pathways involved in hypoxia. What is hypoxia? As you can see that, I don't know why. Okay, so as you can see that in this slide, there are 3 different reactions involving HIF1 alpha, which is a hypoxia-inducible factor 1 alpha, and you don't see an oxidative side of this side of the redox spectrum here. So why I am insisting this? Very importantly, because they studied about the normoxia versus hypoxia, not the hyperoxia. So hyperoxia is connected to oxidative stress that we all know about in several illnesses, chronic disorders, where you see everything is oxidative stress. Truly asking, if you go back and you look at the lab observation notebooks, did we get all results driven, pushed towards the oxidative condition? Or some of the settings, whether you saw the other side, where you have more of antioxidants rather than more of reactive oxygen species. So for this discovery, they received recently Nobel Prize, these 3 individuals, which indicates that it has a very important message, and it matters to study more. So now let us look at the redox spectrum. We all always know about oxidative stress for the past 7 decades or more. But when you look at this schematic representation here, the physiological signaling occurs mainly in the normal redox. Normally, years ago, we believed that redox is uniform across the individuals. But lately, with a lot of basic studies, it has been proposed that redox is not uniform across the board. Within your body, each organ has its own redox makeup. Not only within the organ, but if you speak about a cell, in this context, let us talk about cardiac myocytes. Within a given cardiac myocyte, the redox is not uniform among the organelles present in the cell, meaning that your endoplasmic reticulum versus mitochondria versus nucleus versus cytoplasm have their own redox makeup. So given that very micro environments or the organelle environments having unique redox state, think about you change the redox environment within the organelles, their unique functions will be lost. One example is that endoplasmic reticulum. The primary role of endoplasmic reticulum, we all know that protein folding. So whichever the protein passes through the endoplasmic secretory pathway, they are folded in the ER at the expense of oxidative reactions. So we call it as oxidative phosphorylation, all those things. So normal signaling, while it occurs under the slightly reductive or pro-oxidative, when you go beyond the reductive, little bit towards hyper-reductive, and on the other hand, you go towards oxidative, then it's going to be adaptive response signaling. So there is a threshold, both arms, one can control, and the cells can respond as an adaptive mechanism. Whereas if they go beyond hyper-reductive and hyper-oxidative, that's where, when it becomes chronic, it's going to be pathological. One important point I want to highlight here, while there is a natural defense mechanism exists, and we all know that, antioxidant defense system. So transcriptionally, you can activate the antioxidant genes. I'll come to that in the next slide, how it happens. But we know that the natural defense mechanisms exist to fight against oxidative settings. However, on the other hand, if you have a hyper-reductive condition, as far as my understanding, my knowledge, until today, we do not know whether there is a self-driven mechanism which can prevent the work against the reductive side. So therefore, this is very important to compare extreme reductive and extreme oxidative conditions and see whether there could be a way to come out of or fight against the reductive stress. And before I go to the actual findings, I want to have you to understand how to define a redox state, or particularly the reductive condition. Again, this definition is still an arbitrary numbering based on the redox couples that you see in the cells, like glutathione, reduced versus oxidized form, and it's NADPH and NADP, cysteine and cysteines, the oxidized form of cysteine, and also any other antioxidant, for example, thyroid oxygen, reduced versus oxidative. So if you measure any of these redox couples or all of them, and you see the level is higher than twofold when compared to the counterpart of the control, whatever you have, then you can claim this as a reductive stress. So this was I'm using this discovery to explain how we discovered reductive stress. So originally, in very short, alpha-B-crystalline is a protein, which is basically a heat shock protein, or otherwise known as small molecular chaperone, which its primary role is, you have seen since the morning various talks from our colleagues that the role of cytoskeletal proteins in the cardiac myocytes are very important for the contractility of the cardiac myocyte. And in this case, the role of alpha-B-crystalline is very, very important in response to stress in the heart or cardiac cells, as the alpha-B-crystalline interacts with the Z disk of the cytoskeletal proteins and protect the integrity of those proteins to maintain the cytoskeleton architecture. So what we discovered here is that initially we thought, we made a mouse model of sorry, I forgot about mentioning. So the mutation in this alpha-B-crystalline makes it oligomerization. What is oligomerization? Any protein that is not having proper structure or having mutation can form aggregate by its own. So then this is normally a 20 kilodalton protein, which becomes as big as 800 kilodalton. And think about it, a big stone of protein which is not responding or obeying the protein ubiquitin system or protein quality control system, but stays in the cardiac cells and obstructing the whole physiology and structure of the cardiac myocytes, and it is spreaded over to the whole heart. So in this case, this particular mutation was originally discovered in humans in late 1990s, and this particular pedigree was presented to the clinic not at their early ages, but at the age of 35 and 40 with bigger hearts. They were developing cardiac hypertrophy just because of this single amino acid mutation. So we rebuilt this mutation, human gene, into the mouse heart, and we studied the basics of biochemistry. Initially, we believed that this might have driven by oxidative stress. But surprisingly, these hearts do not have decreased glutathione levels, increased lipid peroxides, and so forth. But what we found was that all of this, what you see here, having increased reductive capacity, which you can see in my next slide, which explains the redox signaling of how antioxidants are being regulated. So in this mechanism, we thought that the role of NRF2, which is the master regulator of antioxidants, playing a major role in the processing of hypertrophic cardiac myopathy in the mutant TG mice. So as you can see that the NRF2 is always complex with the KEAP1, which is the repressor, and it degrades the NRF2 through ubiquitination process. However, in response to signaling or dissociation of NRF2 from KEAP1, can activate the NRF2 and binds to its target genes, promoters of target genes, and wherein you can see the activation of so many targets, including all antioxidants. So you can see that there are GSH producing, utilizing enzymes, proteins, as well as the NADPH production pathway enzymes were activated in this model. So we then developed to ask a question whether activating NRF2, since we saw the NRF2 activation in the R120G model, the mutant protein model, we decided to have a mouse model for unique reductive stress without any mutant protein. In this case, you can see that we have 2 lines of mouse, transgenic mice, where the original mouse with alpha MSC promoter, which drives the cardiac expression, along with the wild type NRF2. In the other line, you can see the second one, which is the constitutively active NRF2. What I mean by that is that in the second form, where you can see amino acid 1 to 89 were deleted, thereby preventing its interaction with its partner, KEAP1. So since it cannot bind to the KEAP1, this NRF2 is sustainedly active. So in this mouse model, what we see, luckily we got several founders, but we selected 2 of them based on their copy numbers, and where you can see we designated them as transgenic low and transgenic high. So you can see the genotype as well as the quantitative, semi-quantitative PCR for CNRF2. So with this, we validated all the antioxidants that you can name. These are, I'm showing just few of them, but bunch of the antioxidants and other anti-inflammatory genes and more cytoprotective genes, anything that has antioxidant response element in their promoter gets activated, and you can see that they are either dose-dependently activated, or some of the antioxidants were evenly activated even at between the low and high, which indicates that depending on their number of promoters and the close proximity of the promoters, that can be differently regulated. Not only the, excuse me, not only the proteins, but also you can see the enzyme activities and total antioxidant activity. Everything is upregulated in a dose-dependent manner in the mouse model of NRF2-TG. Again, to recall, this is nothing but you are simply dumping the antioxidants in the cardiac myocytes. So what happened to these hearts? Oh, sorry, before going there, everybody asks what is the unique marker for reductive stress? And I can ask back another question to answer that. What is the unique marker for oxidative stress? There are several markers for oxidative stress, but it is the balance, right? So it is, if it goes this way, you can say oxidative, and other way, it could be reductive. So likewise, we try to attempt to understand, thank you. Understand whether we can come up with a nice marker for reductive stress. In that effort, you can see that when you have non-transgenic control versus transgenic low and transgenic high, what you see here, even on the non-transgenic control, which is supposed to be the basal level, right? So there you see some level of binding for HNE with the proteins or lipid peroxidation. So which means that when compared to the basal, TG low and TG high are dose dependently down, which means less oxidation is occurring in these hearts. So basically you are preventing the basal oxidation that's supposed to happen in the hearts, which shift the redox towards reductive side. So another proof here, very important this one, because we measure RYS, reactive oxygen species, different probes, but which are not very specific and very reproducible. Therefore, we try to attempt, in collaboration with Dr. Ron Mason from NIH, to inject the mice with DMPO, which is a stable probe that can bind to the oxidized proteins, and you can use an antibody for DMPO and detect the oxidatively modified proteins. Just simply think about non-transgenic itself, you see a very positive signaling for anti-DMPO when compared to the TG low and TG high. So which indicates that you are basically preventing the ROI signaling. Just think about it. If you don't inhale oxygen, what will happen? Something equal to that. So when you see that ROI signals are completely prevented from your cells, then day-to-day life, particularly the cell survival mechanisms, even apoptotic mechanisms, there are so many things are required the ROIs is required for so many different signaling in the cell to make up the normal physiology. So such conditions are being completely taken away. So here is the another way of looking at it. We measured the glutathione levels in these hearts, and you can see that transgenic low and high dose dependently increased GSH levels corresponding to the increased redox ratio for GSH and GSHG. So in this cartoon, we manipulated the CANRF2, basically the transcription factor of antioxidants, to be constitutively active, and thereby we activated pretty much all genes and proteins involved in this pathway, activated or increased at the level of transcription, translation, and functional levels. So which we can call this as a mouse model for reductive stress in the heart. So what did we see in these mice in terms of cardiac output, cardiac function? So in short, I want to show you this. At the age of 6 months, both TG low and TG high, when you compare, there is obvious cardiac hypertrophy phenotype in the TG high mice. And also you can see the histology images, severe cardiac hypertrophy, increased thickness of the ventricle and reduced left ventricle volume. So this is also complemented with the markers of cardiac remodeling at the gene level. And these mice, we found that the TG high mice facing the mortality starting from pretty much after 50 weeks, and we see that about 50% of them, 60% of them died at the age of 80 weeks due to realize this. Dr. Kim has very nicely said that heart-to-heart talk is very important. But I'm talking about 15 years ago, sir. Heart-to-heart talks were really fruitful. But now if I start talking to my students heart-to-heart, it goes different way. So that is a challenge as a PI we do have now. But going back, I was not, I was unable to convince my team, including my mentor, that there exists a reductive condition. So I was blinded after some time with 12 number of animals, and I was completely blinded. Then I had to re-genotype them based on my biochemical assay, not the tail-based DNA analysis. So just with single GSS assay, I was able to segregate the 12 mice, 6 of them into as transgenics and 6 of them into non-transgenics, and they're supposed to they had a highly reductive site. Then the story was discussed by all other collaborators, and we made a decision how to name it. When we went back to the literature to name it, is there a name called reductive stress? We went back. In 1952, a scientist named by Britton Chance has mentioned in one of his reactions, not actually a finding, but he proposed an idea about how a reductive stress can happen. With that, then slowly it grew. There was a long pause until 1995 and 2005, I would say, and then we got a baby in 2007 with an interesting finding of reductive stress associated with the major human heart disease. So what is the age of this heart disease now? I mean, reductive stress now. As I said, maybe 17 years. It's still a teenager. So teenagers like you need to jump into these sort of studies because it's underrepresented here, and there is a lot of things not only happening in the heart, but other organs to be studied for redox mechanisms. So when you look at any pharmacological testing or any intoxication, any challenges, stresses, you should look at both the ends, not just focusing on the oxidative side. If your results are reproducible and you can find something new. So with that, I would like to say that there needs some stress always. There needs some stress. If you don't have stress, then you're inactive. So that the level of stress that you need is eustress. People say it's eustress, and which is required for keeping up your physiology. Let me go back and tell you one small kavidai. People were talking about stress, right? Not only redox stress and other stresses. So it is so simple to control the stress. இப்போ நான் ஒரு கவிதை சொல்ல போறேன். டைட்டில் வந்து February 14. என்ன கவிதைன்னு புரிஞ்சிருக்கு எல்லாத்துக்கும். கவிதை என்ன தெரியுமா? பெண்ணே அன்று காதலர் தினம். பெண்ணே அன்று காதலர் தினம். நான் மலரோடு வந்தேன், நீ மலர்களோடு நின்றிருந்தாய். நான் ஒரு மலரோடு வந்தேன், நீ மலர்களோடு நின்றிருந்தாய். தேர்வு எழுதும் முன்னே நான் தோற்றுவிட்டேன். இனி நீ எனக்கு தோழி மட்டுமே. இன்று முதல் இது நமக்கு நண்பர்கள் தினம். இதில் என்ன புரியுது? What do you understand from this? The moment he realized she is already engaged, he changed his mind to become a friend. Instead of forcing or throwing acid on her face. That is the level of stress. So you have to make it yourself. So other than that is important. You can easily change your mind. I mean there are so many other opportunities. Okay? Hopefully you understood this. So therefore, need more free radicals to control reductive stress. So you cannot eradicate or eliminate the ROS completely from the system, which is going to be detrimental. For example, what exercise does. If you do exercise, you are normally increasing your metabolism. Whatever the oxygen that you inhale, 3 to 4% of the oxygen is converted into free radicals. It's basic metabolism. But we also know that increased exercise is beneficial. How come? Increase the exercise, you are increasing your oxygen inhalation, and you're producing more radicals, and it takes care of your signaling mechanisms. Okay, with that, we have published so many papers, and now you can see that there are about more than 7,000 papers published since the past 15 years. I would like to thank our team. You can see all the postdocs and grad students and undergrads, and also the former mentor, Dr. Ivor Benjamin. Without his kind mentoring and support, I would have not been able to reach this level. And also all my in UAB, within UAB, like Dr. Hindlal is here, and other collaborators across the globe, including collaborators from PSG. And I want to mention at this junction, the bridge for my connection with PSG happened my friend and colleague, Dr. Victor Arogya Das, who is actively working behind the screens. And with that, and I want to acknowledge my funding, and now I'm up for questions. Thank you.
Thanks for that fantastic talk. We have time for a few questions.
Question கேட்கலாம், யாருக்கு எதுவும் புரியலன்னு அர்த்தம்.
Good evening, sir. So I am currently working in a project to inhibit the hypoxia-inducible factor pyrrole hydroxylase 2 enzyme, sir. But I was in dilemma whether the inhibition of this enzyme will lead to reduce the tumor.
You're asking so many questions not related to my topic. Cancer, there are a lot of studies, hypoxia-inducible factor is playing a role in drug resistance and so forth. But relevant to the inhibition, I think there are different small molecules to inhibit HIF1 alpha, but personally I have not used in my lab and I haven't studied. I just showed the slide at how redox is going to be very important. Sorry about it.
Thank you, sir.
Thanks for the presentation, sir. You said about NRF2 gene, NRF2 protein. How can we use this NRF2 in cell death pathways and ferroptosis and
Oh, it's a lot of literature there. So understanding about NRF2, we think that it is protective. I forgot to tell this in my presentation. So NRF2, we think always it is protective. Yes, when you need protection, NRF2 plays a protective role. So for example, there are a lot of polyphenolic compounds from food additives or food extracts can activate NRF2. And there are small molecules. For example, broccoli extract or other sprout extract, they have like components called dimethyl fumarate, sulforaphane, they can activate NRF2. If you have a situation where too much of oxidative stress. But if you activate NRF2 without a problem, at a basal setting, then it's going to screw up your redox system. For example, okay, you are in this crowd. You have a gun in your pocket. When do you open the gun and start firing? When you have offense, right?
Yes, sir.
But if you don't have offense, if you start firing the crowd, then you are offending the environment.
Yes.
Like what? NRF2, under basal conditions, it's not activated. Even for maintaining the basal redox, you do not need NRF2. Only it's a stress response factor. So that's why it's very important. You need NRF2 must be activated only when there is a need.
Is there any chance of NRF2 getting activated in with diseases?
I don't know whether most of the oxidative stress diseases early on, for example, if you have early onset of RY signaling, NRF2 gets activated. But it becomes chronic, then NRF2 system will be impaired. Okay, and another thing you can think about, as simple as like if you do 30 minutes of exercise, fast walking can activate NRF2. Both in, it has been proven in humans as well as the mouse studies that NRF2 can be easily activated through physical activity. Other than that, I have no idea like any other particular disease can activate it.
Thank you, sir.
Welcome.
Good evening, sir. So my question is about, you have stated that oxidative stress is an important thing. So
I didn't say that oxidative stress is important. I said oxidation, ROS is important.
Yes, sir.
But
Reactive
oxidative
oxygen
stress,
species.
a physiological basal level of ROS, reactive oxygen species, or reactive nitrogen species are very crucial for the basal mechanism.
Yes, sir. For those ROS, we have some enzymes to enhance or to suppress those ROS. Whether to increase or suppress reduction stress, we can use enzymes to enhance or slow down those
very
levels.
good point. And if you look back the literature, many of the perfusion studies, even in the cardiac setting, people try to inject superoxide dismutase into the perfusion or catalase, glutathione reductase. I mean, when you try one pathway, what happens if you do superoxide dismutase inject, then it can kill all the ROS. Yes, but the other subsequent mechanisms may not be, subsequently they is forwarded to that. That's why the approach we took here is that NRF2, because NRF2 is global, it can hit all the antioxidant enzymes. Like the dose and duration of activation of an enzyme is very important to see an effect or not to see an effect.
Thank you, sir.
All right, if there are no more questions, let's thank him again.
Thank you.
We are thankful to Dr. Rajashekaran for his insightful session. Now, I request Dr. J. Shankar Ganesh, Associate Professor, CMMT, PSGIMSR, to felicitate Dr. Rajashekaran.
Hello, sir. எப்படி இருக்கீங்க? Nice.
Thank you, dignitaries, for the knowledgeable sessions. Thank you, participants. Let's break for tea and assemble back at 5.
privileged to have a team from the American Heart Association with us today, led by Dr. Suresh P. Damodara, Executive Medical Director, President, American Heart Association Midwest, Florence Wollmel Heart and Vascular Institute, Edgewood, Kentucky, USA. Joining him are the expert team members, Professor Dr. Sachin V. Menon, Consultant Regional Director, AHA, Thrissur, Kerala, Professor Dr. Vishwas Seth, Quality and Certification Consultant, India, AHA, Mumbai, Mr. Shriman Ashok Kumar, Consultant, AHA, Kottayam, Kerala, Dr. P.A. Kumar, Coordinator, Simulation Lab, PSG IMSR, Dr. Arun Samwell, Lecturer in Simulation and AHA Instructor, BLS and ACLS. Let's witness the first-hand power of CPR and its potential to save lives. First, we start with the presentation by Dr. Suresh Damodara, followed by the quiz and a live demo.
So the slides can come up. So when all of you heard a lot of the scientists talk today about heart disease and stroke and how heart attack is something when an artery gets blocked. I want to take a step back and describe 3 patient scenarios for everybody in the audience and the students. So let's say there's a 45-year-old who is now driving his car in Pilamedu and is coming there and starts having discomfort in his chest. And he thinks he had a big afternoon meal and he thinks that's the reason for this discomfort in his chest. He keeps driving the car for a few more minutes, goes to his house, still starts having the same kind of discomfort in the chest. Also feels that it's very uncomfortable for him to breathe. His wife tells him, let's go to the doctor and figure out what this is. And they come to the PSG hospital, and let's say it's about 20 minutes since the onset of the symptom. First, when they come to the hospital, the first thing that happens is, after seeing initially the nurse, they get what is called as an electrocardiogram or an ECG. So that ECG in this person may show that it's really abnormal, he's having a heart attack, meaning that one of his arteries has closed off. This person now, traditionally in Cincinnati, when I take care of patients, 30 minutes into them having symptoms, they come to the hospital, they get an EKG, it gets activated, and then I take that patient to the cath lab, I do an angiogram within half an hour, I will notice that that artery is closed. With the help of a wire, a balloon, and a stent, I would reopen the artery. Within 90 minutes, if you open that artery, it'll be as though the person has not had a heart attack at all. You have saved the heart muscle from dying. Same scenario, this 54-year-old, let's say, goes home now, doesn't listen to his wife, says, no, I'm not going to go to the hospital. 3 hours later, she goes out and comes back, and she finds him dead in the house. This is a very common theme and a very common thing that happens every day. So I'll go forward and talk why cardiopulmonary resuscitation is very important. And this is something the world over the AHA is famous for. C is cardio, which means the heart. P is pulmonary, which means the lungs. Resuscitation is trying to get the heart and lungs to function so that the blood goes to the brain. The problem is not the heart, the problem is actually the brain. Because sometimes when I do the procedure, I open the artery and I find that the artery is open, the heart's okay. The next day the patient doesn't wake up because the brain is dead. I'll have to tell the family the heart's doing okay, but he or she is not going to make it. So that's the value of immediate CPR. How do I change the slides? Do you have the clicker? Okay. So this is my hospital system that I work in. Next. Next. So the second scenario I described, I want to take a few minutes and describe. எல்லாரும் வந்து heart attack ன்னு சொல்றாங்க. Heart attack na என்னதுன்னா, your heart's functioning very well now. 20 minutes later, something happens inside your arteries. A blood vessel closes, and then because of that, suddenly the heart stops. The heart fibrillates because it has no blood supply, and the person dies. That's sudden cardiac death. The two reasons people die commonly. First is arrhythmic death, which means the heart suddenly beats at 300 beats per minute. It goes fibrillating and then stop because it cannot go further because they spoke about the cardiac muscle. After a while, the cardiac muscle won't be able to function. The heart stops. The second type people die is they have a heart attack, the artery closes, their symptoms get more and more, and then at some point in time, the heart completely stops. It doesn't go fast, it just stops. There's two kinds of sudden cardiac death, and you can see that 90% of cases, sudden cardiac arrest leads to sudden cardiac death. The heart abruptly stops beating, and there is barely a few minutes for intervention. And the idea is, let's say somebody's heart stops and you find them going down and you immediately start CPR. 1 in 2 of those patients, if you start CPR, there is a strong chance that something could happen to them that could benefit their heart. But still people ask me this question that, இல்லங்க இது வந்து இந்தியா CPR பண்ணா என்ன ஆகுது, எனிவே ஹாஸ்பிட்டலுக்கு போனோம், ஹாஸ்பிட்டலுக்கு போனாலும் எதனா ஆகுமா? The idea is not that. If you do CPR, there is a chance that that person will live. If you don't do CPR and you wait for somebody else to come, that person's dead. So the idea is, எங்க இருந்தாலும் you immediately initiate CPR. The first thing you do is you go to the person, you try to tell your friends to call for help, then you go, you start CPR, we'll show that to everybody. You make sure they have a pulse, they are not breathing, You immediately start pounding on the chest, 100 to 120 times a minute, and it's as simple as that. You don't need a doctor, you don't need a MD, you don't need a PhD, you could be a science student, you could be anybody. The first thing is you got to notice that the person's down and you got to initiate CPR. Next. So heart disease is the leading cause of death in India. It's the #1 cause of death. Stroke is the #3 cause of death in India. And you can see that #9 is diabetes. 30 years ago when I was in medicine in India, and the #1 cause of death was diarrheal diseases. The #2 cause was tuberculosis. Now the world over, the #1 cause of death is ischemic heart disease, or what we call as traditional coronary disease. Next. So in the US, the average age of a heart attack is over 60. In India, the average age of a heart attack is in the 40s. It's somewhere between 40 and 45 years in India. In the Middle East, it's also in the 40s. So you can see that 1 in 3 patients who has a heart attack, where's Vishwas? Is he there? He's standing there. So I always scare the people by saying 1 in 3 of you will die from heart disease. And that's actually a statistic which is very, very true. So if you're not careful, 1 in 3 persons can develop heart disease over a period of time. So it's extremely common that heart disease and stroke are present. And you can see this in India, the chances of dying is between 40 and 50. People sometimes ask me, so why is that? The reason is, in your 40s and 50s, your heart does not know, it's got normal blood supply. You may have a mild plaque somewhere. So 1 minute when the blood supply stops, the odds of it recovering is very less because it doesn't know what to do. It just fibrillates, people die. In your 50s and 60s, you gradually develop more disease and you develop collaterals to the other parts of the body, and to the heart. So in a 65-year-old or a 70-year-old, they don't die when they have a heart attack. They develop symptoms, they take them to the hospital. Whereas a 35-year-old or 40-year-old is found down, or வீட்டில் யாருன்னா பார்த்தாங்க, நான் வந்து போனங்க அவங்க வீட்டுக்கு நல்லா இருந்தாரு, நான் வந்து திருப்பி வீட்டுக்கு போயிட்டேன், 15 minutesல கூப்பிட்டாங்க, அவர் இறந்துட்டாரு, something like that. So that's a common story that you hear, because a young heart fibrillates, whereas an older heart, because of collaterals, people tend to live longer or come to the medical system and get treated. Next. So the other statistic that everybody needs to know is, why should you know CPR? The reason is, most people who know CPR actually save one of their family members or their friend or somebody that's known to them. So this is something that's very, very commonly forgotten. So the reason to learn CPR is, one day you may save somebody. God willing, it's not your family, but there is a chance that somebody known to you somewhere you could save. So this is very real. If they know the life-saving skill of CPR, they can respond immediately by applying force to the chest to revive the heart. Next. Next. So in India, approximately 5 to 6 lakh people die each year due to sudden cardiac death, and shockingly, a significant proportion of these individuals are under the age of 50. Next. So given only that 2% of the adults in India know how to perform CPR, there's a pressing need for widespread education and training. Every citizen should be equipped with this life-saving skill. For every 1 lakh people, India witnesses 4,500 cases of cardiac arrest, while in the US, only 60 to 100 cases. So you can see the gravity of the situation, that the number of people who have cardiac arrest in India is way more than in most parts of Europe and in North America. Next. So this just shows that there's a regional variation in terms of awareness and likelihood to perform CPR, hands-only CPR. So if you look at Southeast Asia, South Asia, North America, Latin America, the bars basically are saying the awareness. And if you look at India and South Asia, the awareness is very, very less. So one of the purposes of having this conference is to actually let the kids and the audience know that this is something that you in your lifetime will know somebody who may have cardiac death, and who, if you happen to be there, you could save their life by performing CPR. Next. So this is, if you look at the number of people, this is not just India, it's Afghanistan, Bangladesh, Bhutan, India, Maldives, Nepal, Pakistan, Sri Lanka. So if you look at this whole of Southeast Asia, Singapore, Malaysia, Indonesia, this is sort of the belt, what we called as the coronary belt, because there's a high incidence of coronary disease, diabetes, hypertension, smoking. So the number of people, that's why the number one reason people die in India is from heart disease. Next. So community CPR is what is important. That's what we are trying to get everybody. And what do I mean by that is, I think there are not many physicians in the audience. It's you who are the people who are going to save somebody's life if you understand what CPR is. Even though we are showing it today here, it's very simple. All you have to do is to Google CPR, go to YouTube, type Staying Alive, the song. Within 30 seconds, you know CPR. It's not rocket science. It's pretty easy to learn. But we are going to teach you the scientific way of how best to do it. And pretty soon, we'll have a demonstration of that. So really, the way it works is you call for help, you begin chest compressions, you continue until help arrives, and then we can show you some visuals as to how that works. So it's very simple. Ningle vande, if something happens, you're first going to call for somebody. You call for somebody and tell them something has happened. Then you start CPR till as much as somebody is able to. A lot of times what happens is, when you start CPR, some of them are going to either gasp and come out of it, so there is a chance that they would reach the hospital, and in the hospital, they could be saved. If you don't do CPR, even that small chance of that person living completely goes away. There is also data to say that even in the hospital, if somebody's heart stops, you still have a chance of dying even in the hospital where the heart stops, because even then, somebody has to initiate CPR. So initiating CPR is key in terms of any type of cardiac emergency. Do we have the video that will come? No, not exactly. You guys will show it later. Next. So we are collaborating with the All India Institute of Medical Sciences, and the idea is to train students, healthcare workers, and the public in CPR. And as we start training more and more people, the idea is to train the trainer. One person trains the other person trains the other, and then you get a system by which more people are aware about CPR, and hopefully one day a life can be saved. So it's called the Nation of Lifesavers Initiative, and it is along with the All India Institute of Medical Sciences. There are 5 centers in which the AHA is partnered. The idea is this movement can go all across India subsequently. It started with the All India Institute. Now we are doing 4 pilot programs in 4 parts of the country. And the goal is very soon PSG would be one of those programs. And then subsequent to that, the idea is it could become 400 programs or 4,000 programs over a period of time. So I'm going to stop here. Hopefully this was beneficial for all of you. Now we're going to come and show you some of the CPR demonstrations. And if any of the students have questions, I'll be listening, I'll be talking, and they'll get ready and we can start the program. So I'm ready to take questions if somebody has any questions. Yes, அதுதாங்க இப்ப காமிக்க போறோம் கரெக்ட்டா. So easiest answer நீங்க கேட்டதுக்கு வந்து center of the chest, the chest should go up and down. You would get recoil if you want to use this. மேல வச்சு சும்மா you have to literally press that person's chest and you should notice the chest going up and down. 100 to 120 times a minute, that's all. In the center of the chest, you hold your hands and they will show the technique right now when they're doing it. Plus we have some demonstration of what it is. Any other questions? Is it connected? You got it? Yep. Thank you so much. This is Dr. Sathe, Vishwas Sathe. He works along with me. He's a cardiac anesthesiologist from Mumbai. So thank you. Thanks,
Very good evening, everyone. Thank you, DP. And now the next exercise that we're going to do, I wouldn't say an event, it's going to be an exercise, because especially for the ones who are going to volunteer. So what we're going to do is we're going to show you a video about how to perform CPR. Like DP said, it's really not rocket science. It's just basic, getting your hands together and pumping the chest to get that blood flow to the brain. The whole idea of CPR is getting flow to the brain. It's usually not the heart that kills the patient. It's the heart which is not pumping and the blood not reaching the brain. So that's what we're going to try and do over here. So are you ready, guys? Anu? Yeah? Right. So the guys are ready. They're going to show you a video first, and then we are going to get some volunteers up on the stage, and the volunteers will perform the CPR just the way they've seen it on the screen. And we are also going to get them to race against each other. So there are 5 mannequins, and the 5 volunteers will do the compressions, and they will be timed on the screen. All right? You want the volunteers now? You want to show the video? 5 volunteers first. Sachin says he wants the volunteers first. So yeah, 5 volunteers. Right? Girls being the brave ones, stepping up. And yes, there is a prize for the winner. We want 5. 1, 2. The first 5 on the stage get to volunteer. 1, 2, 3, 4, and 5. That's it. Thank you. Thank you. Yeah. Thank you so much. So you all can just grab whichever station you want to be on. Yeah. You want to show the video first? Yeah. You ready? Yeah. Just watch the video once. You can stand on the side so that the audience can also see the video. So this is what you do when you see someone collapse.
If you see a teen or adult suddenly collapse, it is important to act fast. Helping to save a life is easier than you might think. Just start hands-only CPR. The first step is to ask someone to call your local emergency number and to get an AED, or call your local emergency number yourself and put your phone on speaker mode so the operator can assist you. Then get directly over the victim. Put the heel of one hand in the center of the chest and put your other hand on top of the first. Then push hard and fast in the center of the chest until help arrives. It's important to push getting 100 to 120 compressions per minute, which is about the same tempo as the song you're listening to right now. Remember, call your local emergency number then ask someone to get an AED. Then push hard and fast in the center of the chest until the AED and help arrives. Your actions can help save a life.
Most working professionals are finding jobs the wrong way. They see a job post and they hit the apply button, and then they hope they get a call for an interview. If you are serious...
Can you mute that, Srimon? Thank you.
So somebody is searching for a job, Srimon. It was a job, something regarding a job. So, saw the video? Very easy? Okay, we'll make it more interesting. I'm standing here talking to you, suddenly I collapse. What do you do? What do you do first? Call for help. And second? Okay, so we have 5 people. That's me in 5 stations. Start. Yes, call for help. Okay. And there is going to be something projected on the screen. Can you just zoom? Vishwas, tap the screen. Tap the screen.
Yeah, that's fine. I got it.
Yeah. So you can see the ambulances which are going in. Move. So mannequin number 4 there. So she's winning. Carry on. Tired? Next. Come. We need to correct them.
Mannequin 1 hasn't started. You'll need to compress.
Here.
Yeah.
Move towards the side. This and compress. 1, 2, 3, 4.
First and second spot is out. Third spot is for the taking. 4, you can stop.
Mannequin number, you can stop.
You can stop. Who's the second, Sachin? Can't tell. Who's?
Okay.
So
it's done. Thank you.
Okay, stop.
So out of 5, I will survive only on mannequin number 4. So what do we do now? We retrain. Okay, we do it again. One more round. Tired or you want somebody else? Tired? Not tired. Okay. So let's see whether your technique is good. Now come on the mannequins one by one. Whoever wants to do it again. 1, 2, 3. You are exempted. You can come. Yeah. So center of the chest. Yeah. Both hands. Stand by the side. Sit by the side. Yeah. One more. Yes. And you can help the others because yours was the best technique. So now let's just cheer them, okay? Because this is becoming too dry. Wait. Let them start. This you're cheering for me to collapse. Okay, so you ready? So 1, 2, 3, I've collapsed. Come on, go. Mannequin number 4 again, taking the lead. 2 is on second place. Again, mannequin number 4 is leading. No, don't do it. You don't ask, come on. You're too fast, make it a bit slow. Towards the center, over here. Yeah. Again, it's mannequin number 4. Mannequin number 2 is the second place. 1, 3, and 5. Okay, done. So I will survive on mannequin number 4, and mannequin number 2 is, yeah, here. So I know which places I need to collapse, where I should take a decision to collapse. Thank you. So, this was just a trailer. Okay, so who won in the first? Where's the winner? The first time? And the second time? Okay. So the prize is, they will get a free BLS training code that they can use. They can study it online, and they can contact Dr. Kumar over here. He runs the PSG training center, and you can get trained in BLS. And then we will give you the code provided you promise to train 10 more people. Okay? Fine. So any questions? Or only questions? How did you feel? Did you become tired at the end of it? No? Okay. Usually when you're doing, it's just a trailer of how difficult, or one, it's more regarding, you know, the awareness of how CPR is done. The attitude that even if I know CPR, it's not necessary that I'll do CPR when somebody collapses. So if you're trained well, there is a higher chance that you will perform CPR on somebody that you find has collapsed. And DPI has very clearly mentioned that bystander CPR is one of the main determinants. So if not, we'll just project the first slide. We'll give the pin. You're scanning the QR code? Okay. To talk about the rules of the quiz and the prize.
Yeah, so thanks, Sachin. So this is a very interesting game. If you've not been able to download the Kahoot app, you can just simply open on your browser, just type www.kahoot, KAHOOT.com. That's all you need to do, or .it, anyone, okay? They will show it to you on the screen. So if you've not been able to download, you can just Google Kahoot, and it will show you that icon, and you can just log in once we give you the pin. Yeah, so that's the pin. Have you been able to do that? So we're waiting for players. As soon as you log in, we will get the players on the screen. Okay, Rubina has managed to do it. Okay, fine. And it will lock you out once we cross 200. Yeah. So the first 200 will get a chance to play this quiz. 67, 69, oh, well, people are fast. Any AHA instructors here? They will be disqualified immediately. All right, 130, 135, 35, yeah. So we're going to wait another 30 seconds before we start the game. $30 slots left now. 20 slots left. All right, last 10 slots left to log in. All right, 4, 3, 2, 1. That's it. Oh, it's allowing 200. I think we'll stop now. Yeah, okay, fine. Right. So this is the CPR trivia quiz, and we're starting now. All right. So the first question, a drug used in emergencies which was used in Egyptian times for making women look more beautiful. Technical problems, screen. Can we sort that out? So this is your question. Which drug used in emergencies during olden times? And your time starts now. And the hint is, look at the picture. Look at the eyes, rather. I can't give a more definitive hint. 3, 2, 1, 0. Okay. So the right answer is atropine. The drug Atropa belladonna was used during Egyptian times by the Egyptian ladies when they used to meet their counterparts for dinners. And that is, I think that is where the term, the candlelight dinner was probably innovated, because what used to happen is in the dark, your pupils used to constrict. I mean, that's a natural tendency. So to look more beautiful, they would instill that Atropa belladonna in their eyes, and the eyes would just widen up. And that's not my theory. It's Sachin's theory. Yeah. Right. Okay, next question. So, okay, fine. The first answer, the fastest finger was Hobble D Hui. Oh, okay. I hope I pronounced that right. Ratisha, Varsha, Ritam, Vishnu. So you also get, not just when you get the right answer, you also have to get it quick within the time. The first one to go gets the first rank. All right? Okay, next question, Shriman. Yeah. The body of a young lady pulled out from the bottom of the river Seine in France inspired what? The oxygen mask, the bamboo bag, the mannequin, or the life vest? Beautiful young lady was pulled out of the river Seine many years ago.
And the clue is, this is the most kissed face in the world.
Your time ends now. The right answer is mannequin. Majority of you all went for life vest. But the face of the young girl was so beautiful that it inspired Lardell to make a mask of that face, and that is the mask, that is the face that you see on these mannequins right here. Okay? So this is inspired, the face is inspired by that young lady who was found at the bottom of the river. And like Sachin said, unfortunately, it is the most kissed in the world. All right? Fine. So let's see who got the answers. Vishnu, Nandu, Satish, Hana, and Balu. Great. Fantastic. Let's go on to the next question. This is a true or false. American Heart Association was established in the year 1924. DP has already given you the answer. So you got enough time. Everyone knows the answer, I hope. AHA, established in the year 1924. Is it true or is it false? 5, 4, 3, 2, 1. All right. DP, we need to meet these 56. These were the ones who were sleeping when you were talking. Right. So 160 of you got it right. That's true. And that is the reason why we are celebrating 100 bold years. And the plan is to celebrate it on a large scale. This is one of the events that is part of that 100-year celebrations, mass CPR across the world. Right. Next question. Okay, the leaders now are Vishnu, Satish, Nandu, Hana, and Amu. Suba is the highest climber. Next question, CPR is cardiopulmonary revival, cardiac pushing resuscitation, cardiac pulse rescue, cardiopulmonary resuscitation. You have 20 seconds left. I think DP's first slide was the full form of this presentation. Yeah. So 144 got it right. It's cardiopulmonary resuscitation. Thank you. Let's see the winners of this question. Vishnu, Nandu, Malu, Aisha, and Amu. Great. So moving on to the next question. When you encounter an unconscious patient who doesn't respond to shaking and shouting, you will first do what? You will call for help, start compressions, look for an AED, or wish you had attended CPR training. "15 seconds left. I think this is pretty straightforward because Sachin just explained to you a few minutes back. We showed you the video also what to do. So let's see who's got right answers. 182 have got it right, call for help. Has anyone, there are a few people who wish you had attended CPR training. We will share our contact details at the end of this quiz, so you can contact us if you want to go for the CPR training. And I believe we just saw your skills lab today. You all have a fantastic skills lab. Dr. P.A. Kumar is here. You can contact sir to go for CPR training. Right, let's see who got it right. Vishnu, Malu, Aisha, Jakatishwari, and Sham Bansal. Fine. Let's go to the next question. The correct sequence for CPR is 15 compressions, 2 breaths, 30 compressions, 1 breath, 30 compressions, 2 breaths, 15 compressions, and 1 breath. This is slightly, probably difficult for you guys. But let's see. Oh wow, not bad, huh? Not bad. 87 of you got it right. It's 30 compressions and 2 rescue breaths. But when we're talking about hands-only CPR, what we just showed you, that is when, if you're not trained in CPR, the least you can do is give the compressions, and that was the whole idea of getting everyone to do this exercise. So if you find someone who's unresponsive, the first thing you do is call for help and start compressions immediately. Do not interrupt the compressions. All right? Fine. Thank you. And let's see who got the answers right. The quickest ones were Aisha, Malu, Vishnu, Ramya, and Dhanlakshmi. Great. So Aisha has climbed up places suddenly. All right? Great. And there's a prize at the end of this quiz. AED is automated electric defibrillator, automated external defibrillator, automated energy defibrillator, and automated extra cardiac defibrillator. This is commonly seen everywhere. Malls, movie theaters, colleges. I'm sure you all have AEDs around here. So let's see who's got it right. Malu, Vishnu, Dhanlakshmi, M, and Riz. Next question, please. This is a quiz. This maneuver is used to relieve choking, and it is called Heimlich's maneuver, cardiac maneuver, Wright's maneuver, choking rescue maneuver. Time's up. The right answer is Heimlich's maneuver, and 62 of you have got it right. And the last one is there a last question? Yeah. The last one. Let's go to the next question. The last one is true or false. Currently, 1 out of 4 people are likely to get heart disease. This is again alluding to DP's talk. The ones who get it wrong, DP is going to have a personal conversation with each one of them. Gosh, DP. I think that's wishful thinking. No one wants to be in that place, you know. 1 of 4, you just look around, 3 people sitting next to you. So 25% of people under the age of 40 are probably having high chances of getting a heart disease, and that is true. All right, so that's the end of the quiz. Let's see who the winner is. The podium for the CPR trivia is third is Prakatis Balu with 6,240 points, and first is with 6,375 points is Adeline. Congratulations. So who's Adeline? Adeline, are you here? Yeah, right. So Adeline will get a heart code free. Okay, so that's the course, the American Heart Association BLS heart code. You will get it as a prize. Right, thank you. Sachin, to you. TP?
Just conclude, right? Just conclude,
Yeah. Thank you, Sachin, and thank you, Vishwas. Thanks, everyone, for listening and participating. It's wonderful to see 200 people click on these questions. Hopefully, you've learned something, and all the best to all the students here. Thanks to all the participants, to all the speakers. It's been a wonderful day here, and hopefully, all of you have got something out of this. Thank you.
Ladies and gentlemen, as we draw towards the conclusion of our symposium, it's time to express our heartfelt gratitude to our esteemed guests who have graced us with their presence and share their expertise. To honor them, we invite our respected principal, Dr. D. Brinda, to felicitate their guests and honor them. Firstly, let's acknowledge Dr. Suresh P. Damodara, President, American Heart Association, Midwest, Florence Warmold Heart and Vascular Institute, Edgewood, Kentucky, United States of America. Next, I request Professor Dr. Sachin V. Menon, Consultant Regional Director, American Heart Association, Thrissur, Kerala,
Warm welcome to each and every one of you to the valediction ceremony. This event marks the end of an enlightening journey through the realms of cardiovascular sciences, and it's only fitting that we conclude with a sense of gratitude and accomplishment. With heartfelt delight, we welcome the knowledgeable dignitaries to the dais.
Occasion, let's invoke the blessings of harmony and unity with a soulful rendition by the student from the Department of Carnatic Music.
सरस्वती विद्युवती संरक्षतु मां श्री सरस्वती विद्युवती संरक्षतु मां श्री सरस्वती विद्युवती मुरली वीणा गान विनोदिनी संवेदिनी मुरली वीणा गान विनोदिनी संवेदिनी चारु चंद्रहासिनी सरसीरुहलोचनी मुरारी गुरुगुहमोदिनी मुरारी गुरुगुहमोदिनी सत्तात स्वरूपिणी हंसिनी ब्रह्माणी आरत्नवर्णरूपिणी सरस्वती विद्युवती विद्युवती विद्युवती.
Guided by the vision and valuable input of our esteemed speakers, I am honored to invite Dr. N. Krishnaveni, Associate Professor and Head of the Department of Microbiology, to deliver the welcome address.
Good evening, everybody. On behalf of PSG College of Arts and Science, Coimbatore, and Biosciences Departments of PSGCAS, I deem it a privilege to welcome one and all to the valediction of International Symposium on Cardiovascular Sciences 2024. Sevikku unavillada podu siridhu vayittrukkum iyappadum. Meaning, when it is no longer time the listening ear to feed with trifling dole of food supply the body's need. But today we had a packed day of listening from our expert resource persons from the United States with deliberations on cardiovascular research and very thought-provoking insights into the deeper science of heart, and a good food to relish and listen to the expert sessions for the whole day. We are now at the end of the day to the valediction of one-day international symposium. We are glad and welcoming the pillar of support for all our activities, the mastermind behind every move of this symposium, our respected principal, Madam, Dr. D. Brinda, for the valediction of the symposium. We missed you from morning, Madam. We welcome Dr. Suresh P. Damodara, Executive Medical Director, President, American Heart Association, Kentucky, USA. Dr. Shaktivel Sadayappan, from University of Cincinnati, USA, the man behind the conduct of such a wonderful program today. We welcome all the invited guests and resource persons from various universities of USA and members of the American Heart Association who have extended their visit to PSGCAS and have spent their day with us from morning, enlightening all the young minds here. We welcome Dr. Sudha Ramalingam, Director, Research and Innovation, CMMT, PSG IMSNR. Welcome to all the invited guests, heads of other institutions, heads of departments of PSGCAS, faculty members of the organizing departments, and a warm welcome to the delegates of the symposium, all scholars and students who have gathered here today. Welcome one and all.
Leadership is the capacity to translate a vision into reality. We welcome one such leader, our esteemed Principal Dr. D. Brinda, to deliver the presidential address.
A happy and wonderful evening to one and all gathered here. Most distinguished chief guest of the today's program, the center of point of attraction, Dr. Suresh P. Damodara, the President, American Heart Association, Midwest Florence, Kentucky, USA, Dr. Saktivel Sadayappan, Professor of Medicine, Executive Director, Center of Cardiovascular Research, University of Cincinnati, USA, and Dr. Sudha Ramalingam, the Director, R&I, CMMT, PSG IMSR. We have our most eminent, esteemed speakers of the entire day. The invitees who are here from morning, the other guest who has accompanied the foreign delegates, the participants from various institutions. It's roughly around 35 to 40 various institutions. Very happy to see all the participants, the erudite heads of the departments in the PSG College of Arts and Science, the life science departments, and the entire team of the PSG Institute of Medical Science and Research, my dear young mind students, the faculties, the heads of the various departments and other faculties from various institutions. A warm greetings to one and all gathered here. It's really amazing, and it is, I could not express my feeling. I feel it as an immense pleasure in standing before you on this wonderful symposium on the important topic called as cardiovascular sciences. First of all, my deep sense of appreciation goes to none other than our Dr. Saktivel Sadayappan, because without him, big round of applause, without him, this entire program could not have happened. He is the connecting pillar. One fine day, we had an information from Dr. Saktivel Sadayappan that a team of the faculties, a team of the doctors, the team of the renowned speakers from various parts of the country are coming to Bangalore. Then he was so kind enough and he contacted our department, that is the Department of Biochemistry, Dr. Saktivel's wife, Dr. Rashmi, who did his postdoctorate in Cincinnati, where we got a good connection. And before she left, I also told her that find out some new collaborations or some new type of the innovations which can happen between the two universities. One word I said, she made it, and she passed the information to us, and I spoke to Dr. Saktivel Sadayappan, and we WhatsApped, and from that day one onwards, the days it took really 2 months for us to connect each other, and Dr. Saktivel and the other faculty members, Dr. Ramesh, Dr. Victor Arogyadas, and the entire team of the organizing committee members, they took it and they made it happen today. So this type of the fostering the meaningful collaboration, fostering such type of meaningful collaboration is the need of the hour, which we had a wonderful opportunity. It is really a very, very good day for us. It's a beautiful day, it's a memorable day, and all the scientists in one platform made all the young budding scientists to connect each other. So even though I was not present in the morning, I missed the event, but my heart was full here only. My mind and my heart, I could not concentrate because so much effort we have taken, and with lot of effort our entire team has made it, but I could not join. But I know that I heard and I got the feedback that it was an excellent day today. All the speakers, they have contributed their insightful, thought-provoking type of the interactions happened every minute. So we all know that this the Heart Association, they always celebrate this last year in the 2023. So what was the theme is that know your heart, and you have to also use the heart. So this was the theme by the Cardiovascular Association. So many of the people or many of the public, they are not knowing the function of the heart, the importance of the heart, because that type of exposure still it's not happening. So what happens is that dreadful of diseases, especially the cardiovascular death. You know that every year nearing 9 million people are dying out of this severe cardiovascular diseases, many type of the cardiovascular diseases. So this is the right time for us to know about the heart, to study the heart, and also use the heart in a meaningful way. So therefore, we have a wonderful team, and right from the morning, and the last session I was here. So the highlight of today's program is the CPR. Excellently, it went on well. It was a live demo, and a team, I should thank the team of the eminent personalities who were here, and they made it so enthusiastic, so lively. The way they conducted the quiz competition, it was so amazing. No words for me to express. I should thank Dr. Kumar from PSG IMSR for the simulation lab which they have there, and they took lot of the effort, and along with this team from the Delhi, they had a kind enough to come here and conduct this wonderful program. So this sort of the symposium is very important. It connects many people. So the students, as you are all here, you all have to interact with the type of the international speakers who are here and get some meaningful insights. So at this juncture, I once again wholeheartedly want to thank the entire team from various parts of our country who are here, who made this event happen. And also I should thank all the sponsors who all they supported a lot to us. And these sponsors, without them, this event also would not be a grand success. And I also appreciate the organizers, the coordinators, the co-conveners, the organizing committee members, the student volunteers, all have meticulously planned the event, and they were behind the screen working towards it. Their dedication and commitment made us to all connect here and come here. So I also thank Dr. Sudha Ramalingam Madam for being here and accepting our invitation and to deliver the valedictory address. So with these few words, once again, sir, I wholeheartedly thank all of you, sir, all the speakers who have come here, and you have given a wonderful day to us. So with this, I also wanted to say that after this program, after this official valedictory program, we have the cultural event also. So as Krishnaveni was telling, not only we have the ears to hear, we also had a delicious food, is it not? So we have to thank the catering department for their hospitality rendered. They have done tremendously. They have taken many pains and they have made it possible. So you also, I wish all the students to stay back and witness the cultural events. This is specifically done for our delegates, foreign delegates, to witness it. Thank you so much for giving me the opportunity. God bless you.
As an initiator of all innovative endeavors to maintain PSG College of Arts and Science at world-class standards, we are thankful to Madam Principal for having these warm words of greeting and appreciation. Thank you, ma'am. I'm happy to to invite Dr. M. Amravati, Associate Professor and Head of the Department of Hospital Administration, to present the comprehensive report of our symposium.
I wish a very pleasant evening to all. I am immensely pleased to present the report of the International Symposium on Cardiovascular Sciences organized by PSG College of Arts and Science and PSG Institute of Medical Sciences and Research, Coimbatore, in association with University of Cincinnati, College of Medicine, Center for Cardiovascular Research, USA, on 6th March 2024. This has been a flagship event of 2024 of PSG College of Arts and Science. Give a big hand to all the organizers and all the participants. The program started with the inauguration in the morning at 10 a.m. with the prayer song by the Department of Music. The welcome address was delivered by Dr. D. Victor Arokyadas, Associate Professor and HOD, Department of Biochemistry, PSGCAS. And the preamble of the program, the theme of the symposium was given by Dr. B. Ramesh, Associate Professor, Department of Biochemistry. So in his preamble, he mentioned that 27% of deaths in India in 2017 were caused by cardiovascular diseases. The purpose of this symposium is to relay the importance and intricacies of cardiovascular research through international collaboration. The felicitation address was delivered by our secretary, Dr. T. Kannayan. The inaugural address was by Dr. T. M. Subbarao, Principal, PSG IMSR. And the presidential address was delivered by Dr. J. S. Bhuvaneshwaran, Director, PSG, Super Speciality Hospitals. In his presidential address, he highlighted the conventional and non-conventional risk factors of heart diseases. Dr. Kannayan in his felicitation mentioned that his PhD was a research about increasing the golden minutes of cardiopulmonary failure. Dr. Subbarao, Principal PSG IMSR, delivered the inaugural address where he highlighted the importance of this particular symposium and how the participants could benefit out of it. The chief guest address was delivered by Dr. Suresh P. Damodaran, President, American Heart Association, Midwest Florence Woodmart Heart and Vascular Institute, Kentucky, USA. The keynote address was delivered by Dr. Shaktivel Sadayappan, the instrumental person behind this whole event, Professor of Medicine, Executive Director, Center for Cardiovascular Research, University of Cincinnati, USA. And in the inaugural session, a very elaborate vote of thanks was delivered by Dr. L. Uthira, Associate Professor and HOD, Department of Nutrition and Dietetics, PSGCAS, thanking right from the persons, the chairperson of this particular event to the housekeeping people who were very instrumental in meticulously organizing this event. A very big hand to all the participants and all the people who helped in organizing the program. Moving on to the sessions, the first scientific session after the tea break started at 11 a.m. The first session was on heart disease leading cause in Indians. Is genetics or the jalebi? This session was delivered by Professor Shaktivel Sadayappan, University of Cincinnati, USA, and it was chaired by Dr. Shyam Bansal and Dr. Hussain Adelehali. And he was mentioning about the 10 cardiovascular risk factors, the Indian enigma, early risks of heart disease, and how people could be proactive in preventing heart diseases by taking care of their diet, exercising, having proper sleep, and taking care of mental health. The second session was on the immunology of heart failure that was delivered by Dr. Pilar Alcaide, Tufts University School of Medicine, USA, and the session was chaired by Dr. Shyam Bansal and Dr. Hussain Adrihali. And Dr. Pilar talked about experimental approach and ongoing research in immunology and vascular biology. The third scientific session was on fixing the engine without stopping the car, BAG3 and sarcomere PQC, by Dr. Jonathan Kirk, Loyola University, Chicago Stretch School of Medicine, US. And the session was chaired by Dr. Satyamangala Nagaprasad and Dr. Rajashekharan NS. And Dr. Jonathan spoke about how we have to take care of keeping a heart beating. And he spoke about the function of sarcomere in the heart. And post-lunch, the fourth session was on mechanism of fibroblast activation and myocardial fibrosis by Dr. Hen Lal, University of Alabama, Birmingham, USA. And that session was once again chaired by Dr. Satyamangala Nagaprasad and Dr. Rajashekharan NS. Dr. Hen Lal mentioned about the dynamics of fibroblast, and he explained elaborately the mouse model and the experiments that was elaborately carried out in their labs. And post-lunch, in the fifth session, epigenetic control of cardiometabolic disease, Dr. Timothy McKenzie from University of Colorado, Medical Campus, USA. The session was chaired by Dr. Hen Lal and Pilar Alkade. And Dr. Timothy spoke about how mouse model was used to explore genetic aspects of cardiometabolic diseases and the importance of curiosity and the nature of continuous questioning in the line of research work. In the sixth session, immune dysregulation and cardiovascular diseases, delivered by Dr. Shyam Bansal, Heart and Vascular Institute, Pennsylvania State University, USA. It was chaired by Dr. Hen Lal and Pilar Alkade. Dr. Shyam Bansal explained about how we are using science to cure or prevent heart diseases. The seventh session was on role of iron in cardiovascular diseases. It was delivered by Hussein Ardehali, Center for Molecular Cardiology, Northwestern University, USA. And the session was chaired by Dr. Hen Lal and Dr. Timothy McKenzie. Dr. Hussein spoke about his journey to USA after leaving a war-struck Iran. And he spoke about iron and cardiovascular disease and how iron helps in health with the $1 million potential. And he spoke about the impact of iron deficiency and the types of deficiencies that lead to heart diseases. In the eighth session, which was on novel pathways regulating G-protein coupled receptor function in heart failure, which was delivered by Dr. Satyamangala Nagaprasad, Cleveland Clinic, USA. Dr. Hen Lal and Timothy McKenzie chaired. And Dr. Satyamangala Nagaprasad spoke about developing novel therapeutics for heart failure through cardiac pathophysiology. And the ninth session was on atrial remodeling, an early event in proteotoxic heart disease. It was delivered by Dr. Rajashekharan NS, University of Alabama, Birmingham, USA. And the session was chaired by Professor Shaktivel Sadayappan and Dr. Jonathan Kirk. Dr. Rajashekharan spoke about the discovery of reductive stress and reductive stress in cardiac remodeling dysfunction. And after the tea break between 4:30 to 4:45, we had a very elaborate and engaging CPR session by Dr. Suresh P. Damodara and his team from American Heart Association. The participants of this particular symposium have been from 27 institutions from all over India, numbering 415 participants. A very big hand to all the participants who have come all the way to benefit from this wonderful, very interactive symposium on cardiovascular sciences. Thank you.
Thank you, Madam, for highlighting the key discussions, learnings, and achievements of this academic amalgamation. We are privileged to invite Dr. Sudha Ramalingam, Director of Research and Innovation at CMMT, PSGCAS, to deliver the valedictory address.
A very good evening to all of you. How are you all? How was the day? It's soon after tea break. You should be a little louder. How was the day? Good. Okay. So first of all, I would like to thank Ma'am Brinda, Dr. Sadayappan, and Dr. Suresh Damodaran for having us here, partner with this momentous occasion with the PSG College of Arts and Science to be part of this event. The greatest scientists are also greatest dreamers, constantly envisioning new possibilities and exploring uncharted territories of the mind. I didn't say this, Jane Goodall said it. Today, we had one such opportunity to envision a momentous occasion where great minds came together to dream, to discuss, and to delve into the depths of cardiovascular diseases. As a public health physician, I am concerned about the global rise of non-communicable diseases, especially cardiovascular disease. Already you might have heard since morning this statistics because it is quite alarming and worrisome, because in developing countries, the major cause of death is cardiovascular disease. More than 1/4 of the population or the deaths is because of cardiovascular diseases. And why? Especially in India, they say there are 6 major transitions that are occurring. One, the epidemiological transition. As Dr. Suresh mentioned in his earlier slides, some 20 years back or 30 years back, the major cause of death was cholera or acute respiratory infection or tuberculosis. But now, there is a big paradigm shift towards non-communicable disease like cardiovascular diseases, diabetes, and cancers. So that is the epidemiological shift. There is a demographic shift. The aging of the population and the lifespan, increased life expectancy because of improved health care. So there is a demographic shift, which is again causing a rise in cardiovascular disease. There is a nutritional shift or nutritional transition. We all know it. How many of you order Swiggy almost once a week at least? How many of you eat out? Yes, I see only one hand, which is not true. Yeah, or daily, more than once. Okay, I see many hands now. So that is the nutritional transition we are talking about. And there is an environmental shift. There is a worsening air quality, which also contributes to the cardiovascular diseases. And there is a socio-cultural and economic shift or transition because of sedentary lifestyle, less physical activity, and online consumerism. Amazon, Swiggy, right? So many other platforms where you can order when you sit in the comfort of your rooms. So these are the different transitions which are attributed to the rising cardiovascular disease pandemic, I would say, in the world. The risk factors and metabolic programming starts in utero. We all think it starts like, when you start eating out or when you reduce the physical activity. No. It starts even when we are in utero, which means even when we are in the mother's womb, the risk factors begin because the maternal weight, maternal nutrition, and maternal health status contributes to this and continues throughout the life. So it requires an interdisciplinary and an interprofessional approach throughout the life course. It is not enough if I start exercising when I'm 40. We have to start as early as possible. The lifestyle changes need to begin as early as possible, and it has to continue throughout the life. So in that context, this event is well timed and planned to cover a whole range of topics, including immunology to epigenetics And I congratulate the entire team from the US and also PSG for putting together a well-structured program covering all aspects of CVD, which can be translated from bench to bedside, and I would say to the community at large. I also thank the team for giving us an opportunity to be part of this wonderful event. As we come to the end of the day, I would like to stress on few things. I'll be failing in my duty if I don't say this. Dr. Damodaran mentioned the significance of addressing sleep and stress. I cannot agree more. It is very important to carry out cutting-edge research, but it is even more important to take care of ourselves. So following the 6 pillars of lifestyle medicine, the diet, physical activity, sleep, not during the class, right, or not during the conference, but as you go back to your rooms, managing stress, and avoiding substance abuse. And the most important one is the social connectedness. That's a very important pillar which we talk very less. We often talk about diet and physical activity, but we don't talk about social connectedness. In this era of gadgets and screen times, I urge all of you, especially the younger generation, because I see the demography is towards the younger generation, to limit your screen time and increase your social time with friends and families. As Mahatma Gandhi said, be the change you want to see in this world, right? If you want your community to be healthy, first you have to follow the healthy lifestyle practices so that you can preach and teach others. I'm sure the day has been an enlightening and inspirational one, and I'm sure we can carry the ideas generated forward and foster collaborations to benefit the community at large. Whether we are basic scientists or public health professionals or cardiologists, our goal and cause is one, that is health for all. So let us join hands to build a community of practice towards this. Jai Hind and thank you.
Thank you, Madam, for inspiring us with your words of wisdom. As we come to the pinnacle of our ceremony, it's time to recognize the dedication and hard work of our participants. I'm honored to invite our respected principal, Dr. D. Brinda, to distribute the certificates to our deserving participants. Let's start with the winners for the best poster presentation. The first prize goes to Ms. Vani and team from Sri Ramakrishna Institute of Paramedical Sciences, Coimbatore. The second prize goes to Mr. John Vinod and team from Sri Ramakrishna Institute of Paramedical Sciences, Coimbatore. The third prize goes to Ms. Subiksha and team from PSG College of Arts and Science. We also have the certificate of appreciation for the poster presenters. I call upon Ms. Gracie Mohan and team from SRM Medical College and Research. Roshni R and team from PSG College of Arts and Science. Mr. Naveen Kumar and team from PSG College of Arts and Science. Mr. Sai Shri Krishna and team from Shri Shakti Institute of Engineering and Technology. Ms. Tarshimi and team from Sri Ramakrishna Institute of Paramedical Sciences. Thank you, dear participants. Congratulations, winners. Your presence and participation has been instrumental in making this event a success. As we near the conclusion of our symposium, we invite you to share your thoughts, suggestions, and feedback with us. Your input will guide us in enhancing our future endeavors and improving the overall experience for all the participants. Dear students and participants, please feel free to share it with us on this platform. Your feedback is highly valued and appreciated.
Good evening, respected dignitaries, and good evening to all the participants. And this one day international symposium was very useful for us. Myself, I'm Shobya from Avinashalingam University, and I'm doing my masters, and I'm the first year of the masters, biochemistry, ma'am. And this international symposium has we have collected lots of information and lots of efforts and how to present. We have learned lot of things in this symposium, and thank you for all the dignitaries who shared and who supported, who cleared our doubts, and thanks for all the participants, and thanks for all the teachers who have coordinated this well. Thank you.